10 minutes.
10 minutes, 10 minutes சொல்லிருங்க.
Sorry, start பண்ணிடுவாரு.
Good morning, students. Good morning. Yeah. So thank you very much for attending this function. I'm very happy that even undergraduate students are also part of this particular function. And just to brief, because yesterday UG students were not there, but this particular function is all about promoting student innovations. And initially we have started with postgraduate research. Students who have done some great innovations as part of their MSc and PhD program. So what we did is we picked around 100 innovations from students, and those innovations, around 70 innovations have been displayed in the Innovation Expo. UG students, have you visited that Expo? Yes or no? Then why don't you visit today? I'll ask your dean just to permit you. Have a look, because this is a good opportunity for you to see your own students have done this excellent job. So we have displayed that innovation, and from that the whole idea is innovations which have got some direct impact with industries or companies. So the innovations which can be upscaled or which can be further fine-tuned with the help of industries, because at the end of the day, whatever the innovation we should do, it should reach to the farmers. That is the ultimate objective of any research what we do. So with that idea, just we have just invited many industries. Yesterday many industries participated. Today also a lot of industries are online. So around that 70 innovations, we are giving opportunity for the young innovators, like the people who are sitting in the dais. We are giving opportunity for them to present their innovation. And this particular presentation is also sent through online, and many industries can view this innovation, and they can come forward to help the students to take it forward. So in that particular process, today I think we have two sessions. Almost close to 6 or 7 students will be presenting their research, and today is the crop, which category? Crop improvement? Today is the first session is crop improvement category, followed by post-harvest processing. So I think your friends will be presenting their innovations. So let us give a big applause to these 3 young innovators. Right. And only one thing actually I assure, this day will be highly useful to you. Whoever assembled here, this day will be useful, because very shortly 2 leading industrial people, they are going to come and they are going to make a presentation. One is Mr. Sachin, and he is the one who is a leader in hydroponics. He is from Hyderabad. And I think he's number 1 hydroponics cultivator in South Asia. Such a big person, and he's on his way from airport to the university. Probably very shortly he will arriving, and he will be making a presentation. And also he is doing a lot of work on herbal pharmaceuticals. That is the first one. The second one will be Mr. Prashant, and who is doing a lot of research on disruptive technology, or I can say that emerging technologies in agriculture. When we talk about emerging technologies in agriculture, what are the emerging technologies in agriculture? Any idea, students? One technology, can you tell? Emerging technology? Yeah, that's right. Artificial engineering, artificial intelligence, nanotechnology, IoT, big data analysis, all are emerging technology. So the second session, the special lecture will be provided by Mr. Prashant. So I think the day will be certainly enterprising to you, and whatever the time you spend, I think it will be more useful. That's the only promise I can give at this stage of time. I don't want to take much of time, because your young innovators are ready to present. Now I leave the stage to Dr. Prabhu, and who is actually coordinating the entire student presentation. Prabhu, please take over. Thank you.
Thank you. So good morning, ladies and gentlemen. crop improvement is essential to meet the demands and the changing world because of climate change and also the land shrinking and population increase and all that. Of course, TNA feels proud to evolve close to 870 varieties for the past 50 years. It's a record achievement. Of course, our technologies have reached far away, and overseas also we have reached. And in this context, there is a lot of ways of developing and improving by using the advanced technology, which will be really innovative as far as students are concerned. We've got 3 musketeers here. They are going to excel exceedingly well. And the first and foremost is Niveda, who will be talking on the artificial microRNAs, mediated resistance against peanut bud necrosis virus in tomato. It's a mediated gene and is randomly permulated sequence also, single standard thing. She'll be able to explain this very eloquently and clearly for the audience. And this is supposed to be the radical innovation. Now, the stage will be for Niveda for presentation for 10 minutes, short and make it sweet. Thank you.
Good morning to everyone present here. Myself Niveta from Department of Plant Pathology. I'm here to share my idea on artificial microRNAs mediated resistance against peanut bud necrosis virus in tomato. We knew that tomato is one of the most important and irreplaceable vegetable in almost all daily part of our family diet. Globally, the major tomato producing countries are India, China, US, Italy, Turkey and Egypt. And India is the second most important country in producing tomatoes, and nearly 50% of the Indian states are involved in producing tomatoes all around the year. For the past year, we had a production of about 2.03 million metric tons in a land area of 8 lakh hectares. Even though we are high in production, we are lacking in productivity. According to statistical data from IndiaStat, we have found that the productivity for the past 3 years is constantly becoming reduced because of it may be because of the biotic and abiotic stresses. Coming to biotic stresses, nearly 50% of the yield loss occurs due to the combination of insects as well as microbial attacks. As a pathologist, I mostly concern on diseases. So, the major diseases of tomato contributing to yield loss include early blight, late blight, bacterial wilt and tomato leaf curl and tomato spotted wilt. Actually, this tomato spotted wilt can cause an yield loss up to 80 to 100% in the severe stages. Actually, this tomato spotted wilt is caused by a virus called as peanut bud necrosis virus. This virus is relative to the death-dealing coronavirus, but you don't worry about that. This virus can affect only plants but not human beings. This virus alone cannot enter into the host plant, so it takes help from insect vectors called thrips. These 3 thrips families are mainly involved in transmitting these viruses. This virus and thrips, this is a deadly combo. When it is present in a tomato field, it will lead to diseases. It will lead to the tomato spotted wilt disease with symptoms like necrotic ring spots, stem necrosis, drying of petioles and flower buds, ring spots on fruits, and early wilting of the infected plants. Actually, this thrips can transmit virus even in the early stages of the plants, but the symptoms are visible to the farmers only at the later stages. So, the farmer will have a dual penalty in case of yield loss as well as expenditure loss. So, for managing this virus, we don't commercially have any viricides. So, alternatively, we go for controlling this thrips. Commercially, we control these thrips with the help of insecticide sprays. But with the repetitive use of insecticide sprays, this thrips will develop resistance at one point. And also, we are polluting the environment with these chemical sprays. And we also making it hazardous to flora and fauna present near the plant ecosystems. And really, it is unsafe, risky to human beings. So, to sort out these problems, we need to find an alternate strategy. Can you tell me what is the alternate strategy for this? That's nothing but transgenics. This transgenics is not only, this is not a new approach to us. Actually, it is an already proven and successful one. For example, the papaya, transgenic papaya, which is resistant to papaya ring spot virus, is commercially produced in Hawaii in the year 2002. But till now, it is an commercially ruling papaya variety, and this technology is also replicated in countries like Bangladesh, Thailand, Philippines, and Malaysia. What we do is, we replicate this technology to ourselves to control this disease. What we here do is, we just make the plants themselves to resist the virus without our help. That is, we just impart or give an artificially genetic memory about the virus to the plants to oppose the virus. In our research study, what we will do is, we will isolate the RNA from nucleic acid from the peanut bud necrosis virus, and we identified and conserved region that is nothing but an unaltered region in the peanut bud necrosis virus. What we do, we identified that conserved regions, and we designed a microRNA that is complementary to this conserved region, and this region is called as an artificial microRNA. What we do is this, we transform this artificial microRNA to our tomato plant, and whenever this artificial microRNA present in the tomato plant, will identify its complementary viral RNA, and whenever virus enters into the plant, it will find them and gives an alert to the plant about the entry of the virus, and it will also start to fight against the virus and will kill the virus at the point of entry. So thereby, this artificial microRNA technology will make the plant remain healthier forever. So the impact of my innovation is, at the end of my research, I will be involved in developing peanut bud necrosis resistant lines. This artificial microRNAs can also be designed not only for targeting a single virus, we can also target for many viruses. That is what I mean to explain is, we can develop a single transgenic resistant tomato plant that is showing resistance to many diseases. As well as we can design artificial microRNA to resist more than all biotic agents, that is insects, pests, and virus, fungus, etc. So when we take this plants to our field, we can minimize the use of insecticides. Since we can reduce the chemical sprays, we can assure a national security in terms of both quality and quantity. Since the viral attacked plants are very inferior in quality, when we develop a tomato plant by this technology, that is transgenic technology, we can have an healthier and healthier tomato plants. And the next is about this regarding the scope of the innovation. Actually, when we adopt this technology to other companies with high yielding varieties, we can combine an disease resistant plant with an high yielding plant. So we can make the plants durable for long lasting periods. And this, by exploiting this technology with the other crops, we can produce many biotic resistant lines. So now it's your turn to take up the chance. Thank you.
Thank you, Nibeta. So we have a lot of research have been going on in clonal propagation, especially in Forest College Research Institute. Of course, we are rapid in in vitro generation also. Our direct organogenesis work is also going on, biotechnology, explant and all that. A lot of work is going on in Tani. But what she is going to do is, she is going to develop a protocol, multiplication of yellow germplasm. She would be concentrating on the good health for lycopene, lignin, tannin and indole. And she'll be expressing the positive properties through her effort. Now we have Janani on stage, who will be talking about micropropagation of moringa for increased and bioactive compounds production. Of course, this will be product innovation. She comes from Department of Vegetable Sciences, Periculum. Now the stage is for Janani to talk a few points on this innovation. Thank you.
Good morning everyone present here. Myself Janani Tangwell, doing third PhD in Vegetable Science from Periculum campus. Actually, many crops are doing in plant tissue culture industry. I attempted moringa in tissue culture for increased and bioactive compound production. Developing a basic protocol for extraction of bioactive compound production is my type of innovation. What's the significance behind it means within a short period of time we can achieve mass multiplication under controlled conditions. It can be taken and we know that people are very cautious about health conscious and for taking plant-based compounds. It shows that almost 80% of the world population depends upon the plant-based products. It can be easily achieved to plant tissue culture, and bioactive compounds from plant tissue culture compounds also easily achieved. If there is any rare genotypes in moringa means we cannot exploit the mother source. So from the mother source a small part can be taken as a explant without affecting it. Micropropagation, callus culture and cell suspension culture, these techniques helps to develop basic plant-based bioactive compounds for pharmaceutical industries, cosmetics and for flavor development. What's the impact in it means actually it's developing under a controlled conditions mean we can the quality can be easily reduced, and by consuming a plant-based product it can be we can have a healthier lifestyle by minimizing the intake of synthetic drugs. Plant-based bioactive compounds have a scope in global market that in 2017 it have a 29.3 billion and in 2021 it have a 39.2 billion with the compound annual growth rate of 6.1%. And when compared to other countries, India have many plant tissue culture laboratories. It helps to give a good future for who are going to develop a laboratory. When Allied Market Research conducted a survey, and it shows that global plant tissue culture market is expected to reach 895 million in 2030 with a compound annual growth rate of 8.5%. For developing plant tissue culture laboratories, there are many agencies to support for finance support, and Ministry of Agriculture helps to give 21 lakhs for public sector and 10 lakhs for private sector. If a farmer have an idea to develop a tissue culture in a small scale means with the help of cooperative society they can get a loan from small farmers agri-business consortium. They give loan up to 50 lakhs. And APIDA, we know that APIDA helps in exporting. APIDA helps to transport plant tissue culture plants in 25%, giving 25% subsidy in the airports of Bengaluru, Mumbai and New Delhi. In India, there are 6 national centers for controlling virus control and quality control. It can be easily diagnosed. Indian Company Act in 1956 developed a biotech consortium, India. It helps to develop a technology and syndication of funds, project consultation, dissemination of information for developing a plant tissue culture in what crops and what we have to do for it, and giving a training manpower. We just need a small interest in it means we can develop we don't want any degree or basic skill means just we need an interest to develop a plant tissue culture laboratory. We can easily with the support of these agencies we can helps to develop a plant tissue culture laboratory. And many crops I like to conclude that many crops are dominating in plant tissue culture like banana, bamboo. They are exporting to many countries. Likewise, why cannot we do moringa? That's my question. That's it. Thank you.
Thank you. Now, anything will be a contest competition mode. So her work started way back in Germany, now spread to many countries across the world. And of course, will be a delicious darling for everyone. It is again akin to oyster mushroom also. She'll be talking on the Pleurotus cultivars. And now we call upon Shivadashna Priya, Department of Plant Pathology of Majra, who will be talking about intra and interspecific hybridization for developing elite Pleurotus cultivars. Of course, you've done a lot of work. Of course, you'll be getting a lot of ideas through innovation. It is a sort of incremental innovation. Now, Shivadashna Priya will be talking for 10 minutes regarding our innovation. Thank you.
Good morning, all. First of all, I thank to give this opportunity to present here. My research work is on intra and interspecific hybridization for developing elite Pleurotus cultivar. So I know everybody eat mushroom, right? Yes? Yeah. So here we are going to improve the strain. That is that nothing but strain improvement. So let me see. So the current scenario is, we all know button mushroom is mostly consumed all over the world. So next to the button mushroom, the second most widely consumable mushroom is Pleurotus. So the total mushroom production in India is approximately 0.13 million tons. So it is nutritionally as well as therapeutically beneficial for the humans. So my utmost aim is to beneficial to the consumers. That is our preliminary. Whatever the works we are done, its main aim is to the consumer preference. So oyster mushrooms, it is ubiquitous in nature. So cultivation of oyster mushroom is also very less. That is, the infrastructure facility is also less compared to the button and shiitake mushroom cultivations. So my impact of the innovation is the Pleurotus mushroom cultivation is mostly influenced by the long cropping duration. That is, it takes up to 50 days to complete the cropping cycle. So that is a one disadvantage. And the second one is late flushing with a low yield. So if the flushing period is late, means the farmers won't prefer to cultivate the mushrooms. That is the second one. And the third thing is reduced shelf life. So anyway, when the consumer is buying the product, means at least he want to store for at least 2 days. So that is a major drawback. So in order to overcome these three difficulties, so my study is to produce a new strain of Pleurotus through somatic hybridization, so which is also called as hyphal anastomosis, in order to produce a hybrid having high productivity with a short duration with enhanced health benefits. Okay. So here we are taking the 4 different varieties. The one is Pleurotus jamar, variety isolate woody one. So it is we got through natural selection. In this, all the characters are there. That is, short duration will be there. Then with good yield will be there. And also the margin is wavy with a smooth. Everything will be there. But the major problem is the brittle in nature. That is, it is leathery and palatable is very low. So even though we are producing a variety with a short duration, high yield, the acceptability won't be there, means there is no use. So this is the drawback in the isolate. The other 3 varieties which is commercially available in even in our department, so it is highly consumable, but the major problem is the long cropping duration. That is, it takes nearly 50 days to complete the entire cropping cycle. So this is a mycelial phenotype and fruiting bodies of different Pleurotus species, that is Pleurotus jamar, isolate woody, and Pleurotus jamar variety MDU1, Madurai 1, then Pleurotus florida and Hypsizygous almarius, variety Kotu. So this is the phenotype of the woody one. We can see it is wavy in nature, whereas the other 3 are smooth and soft. The major problem in woody is that the less palatability. The other 3 is long cropping duration. So why somatic hybridization? This is a simple flowchart to explain you. So here we are taking the parent A and parent B. For example, the parent A, it's a woody. So it has short duration, early flushing, good yield, but the problem is poor palatable characters. Whereas in parent B, it is a long cropping duration, but it is having good yield and high palatable characters. So we are going to cross these 2 in order to get a hybrid having shorter duration, early flushing, with the high palatable characters, and also with enhanced shelf life, higher bioefficiency, and with increased nutraceutical properties. So this is our study. So let us see the components involved in the somatic hybridization. It is just simple. We are going to collect the spores from the basidiocarp, and we are diluting the spores in order to get the single spore, because we are going to cross the 2 parents. So 2 spores are to be crossed. So we are going to make the individual spore isolation by spread plate technique. So from that, fast growing monokaryons are isolated and then the master plate is prepared. So from the master plate of two parents, each spore is crossed by using dual plate technique. So if the parents are compatible means there will be the formation of fluffy growth is formed at the junction of the two plates. Here we can see the compatible mating type. So from the fluffy, if we are not sure, we have to analyze again means we can see through microscope that is the formation of clamp connection which indicates the formation of hyphal anastomosis, that is somatic hybridized. So from the fluffy region, it is again subcultured and mother spawn is prepared, which is nothing but the seed for the preparation of the mushroom bed. So from the mother spawn, bed spawn is prepared, thereby it leads to the formation of hybrid. So the hybrid will have the characters of both the parents. So whatever the parents we are taking, the both characters will be there. So we want the hybrid with a short duration, with a good yield, with a high palatable, with increased nutrient properties, which is beneficial for the consumers who are going to consume and beneficial for the farmers who are going to cultivate. So this is the somatic hybridization between the monokaryons. We can see the compatible and incompatible mating types. So incompatible mating, there won't be any fluffy regions in the interjunction of two parents. Okay. So the present research is to develop a hybrid with a shorter duration, high yield, increased shelf life, higher bioefficiency, and also assessing the nutraceutical properties. If we develop a hybrid, we will go for the development of value-added products. Thank you.
Thank you. For other themes, now we have theme 4 on the post-harvest processing. Okay. We'll have an interaction session now. You are expected to ask questions and they'll be able to answer. Now the question answer session. Please come to the front and receive the questions.
Students, it's an opportunity for you to interact with the student innovators. So feel free to ask questions. Because you know that what they have done already, all the three, they have presented their innovations. So now it is up to you. It's not, it's just a discussion. Take it as a discussion. Even you can also get something how to plan your research also. So I think consider this as an opportunity to discuss their innovation.
No more queries among the audience? Huge audience. Ma'am is there. Please. Definitely.
I have a question to the mushroom girl.
Ma'am, thank you.
I just wanted to find with you at what stage are you in and when would you complete your research? And then when can you come back concretely to say that the one that you are actually working on will be a commercial product? How long is the duration? And I also wanted to explain to you that you actually presented quite well in terms of the journey. But if we are a commercial company and we like your research, what you're doing, and we wanted to kind of fund and take it forward, what is your thought process?
Okay, so far we have developed two hybrids between the Jamar Woody 1 and PF. So we developed two hybrids. So we are testing that nutraceutical properties and also we are comparing with the parents. So we will develop up to the hybrid process. So then if we develop a hybrid that is better than the parents means, then the industries will join and we can make the value-added products, which because even nowadays the consumers are ready to eat. So not to buy, cook and eat. If we made value-added products means it will be useful for the consumers. The product which we made will be anyway better than the parents. So it will be beneficial for them. So far we developed two hybrids. The ongoing process going on. So within a year we will develop. Within a year we'll develop. So we are testing. All of a sudden we can't tell it is a hybrid. We have to test it again and again. So testing is going on. So we will develop within a year.
So will you be able to work with your college to make this project successful and also commercialize?
Yes, ma'am, we can.
Would you want to become an entrepreneur to do this?
Yes, ma'am, we can.
If there is no question, to wind up. Congratulations. Okay, please.
Question is for you only.
Thank you, sir.
So you have mentioned the characteristics of a parent A and B, but none of the parent you have not mentioned about the shelf life of that parent. So you have given in the outcome that you will develop with a longer shelf life hybrid.
Yes.
So what is the existing parent shelf life, and what about what you are expecting in that?
Actually, sir, so far we developed the hybrid. The shelf life is somewhat 1 or 2 days only. This differ from the parent. So if for example, if the parents having 2 to 3 days shelf life, means the hybrid having 4 to 5 days, like that only. So, but what we are going to explain, what we are going to do is, so we can test it again. If the hybrid, if it is better than the parent, only 1 or 2 days only shelf life is differing. So we can't tell, before developing a hybrid, we can't tell this is the shelf life of the hybrid. We are telling this is the particular shelf life. We can't able to tell correctly. So the research is going on. We will tell when the hybrid is developed, we will tell this will be the shelf life difference of parents and hybrid. So far we are developing only 2 hybrids from a single cross. So if you are doing 100 to 200 crosses, from that only 2 hybrids we are obtaining. So process going on, we will tell at the last.
Okay, I have one question for everyone. So how many targets you have chosen for your microRNA mediated, is it RNA interference?
Yes, sir.
Okay, how many targets you have chosen?
So far I have done for only 1 target, sir, but have chosen 5 targets, sir.
Will it be possible for you to reveal which target you have chosen?
Sir, for peanut bud necrosis virus, that LRNA region, that is that LRNA is, which is responsible for replication, I have chosen that, sir.
Thank you. So the second question is for moringa, moringa tissue culture.
Yes, sir.
Is tissue culture for moringa needed? You can grow easily, right?
Yes, sir.
Then why do you
I
go
just
for tissue?
attempted my work in that.
No, I understand, but the question is a simple question.
Yes, sir.
When you can grow moringa by cuttings, why do you go for tissue culture?
So actually, moringa is rich for its therapeutic properties. If we have one source or we have rare genotype which are in a high yielding or a high therapeutic value, means we cannot go for breaking. Actually, moringa is limb cutting. We have to use for 2 meter of moringa, taking 2 meters. If we exploit the mother source, means we cannot furtherly generate it. That's why I taken moringa for that tissue culture.
Okay, thank you. So your research seems to be interesting, and this is the first time I'm seeing a hybridization in fungus personally.
Okay, sir. Thank you, sir.
How do you confirm that whatever you get is a hybrid?
So first of all, while doing dual plate technique, we can see the clamp connection as of initial observation. That is the mating, fluffy growth will be there at the center of the mating of 2 parents. The next one is we can observe phenotypically. While hybrid is produced means that will be different from both the parents. So we are selecting the 2 parents that is morphologically dissimilar. So when we cross that, and the hybrid will be the combination of both. We can observe phenotypically. That is the one more thing. And next one is molecular analysis, sir. So phenotypic observation is the first thing. If we are selecting wavy smooth, means the hybrid will be both the concern, not that much wavy, not that much smooth. It will be like that, sir. That is the first observation. We can confirm that is okay, this is different from the parent. So we can go for the next step, sir.
No, I learned I know some amount, some of plant genetics.
Okay.
Does recombination happen in your hybrid? Have you ever observed any recombination events?
No, sir, not yet.
No.
It's still going.
So did you attempt any protoplast fusion?
No, sir. Protoplast fusion is done in mushroom hybridization. In intergeneric, it is done. So for intraspecific and interspecific, we are doing somatic hybridization only, sir. So we are trying this method to attempt to develop a hybrid, sir.
Okay, thank you.
Thank you, sir.
So this is a great advantage of cross learning. Just can you see that how much of ideas she is getting? She's in the process of her PhD program and innovation. And a lot of ideas she is getting means the final product will be the best one. When you get that ideas in between, now she has the confidence of facing the crowd, answering the queries, getting a lot of inputs from scientists like him. So I think all these things will actually enrich her knowledge on that particular topic. And finally, what the end results will be a product which is ready for market, or a product which has got high success rate. So that is the whole idea of actually bringing all the people and connecting these people with scientists, and as well as Madam has asked some nice queries and also suggestions. So all these things at the end of the day helps for a word called as cross-learning. When we discuss with different people, that's the whole idea. So thank you very much. Now I think I'll leave the stage to Dr. Prabhu again.
Thank you all.
Thank you all for handling the questions very intelligently and very calmly. Now the fourth theme is very important theme, post-harvest processing, because you all know that post-harvest losses are very dreadful, and it clears the exchequer to the core, and a lot of deceptive colors are coming to the scene, and a lot of MLSP are coming, which are not so conducive for them. The children, health, all are affected because of the coloring agent, which are very deceptive, and synthetic colors are also coming in all that. Now these people want to go a natural way, so we got anthocyanin, natural anthocyanin food colorant from Rosale for confectionaries, which is very attractive red color. It is a disruptive innovation. Of course, we have got a lot of Netflix, Prime videos are disruptive. Sindhu Bharati will be also bringing a disruptive innovation come product innovation. Now we call upon Sindhu Bharati to talk on the natural anthocyanin food coloring from Rosale for confectionaries. All the three can come and occupy the stage. Yokeshwari, where are you? We have been calling you for the past 10 times. You're not attending a call. Yokeshwari is here. Riyan.
Good morning to one and all present here. I am Sindhu Bharati, PhD Vegetable Science from HCNRI Perikulam, with the guidance of my chairman, Dr. K. Naheshwari, Professor, Department of Vegetable Science. Today I am going to present on my work, Natural Anthocyanin Food Colorant from Rosal for Confectionaries. My work is on Technology Readiness Level 2, and it comes under Disruptive and Process Innovation. Color. Color is the first thing we all notice when we start eating a food. It is the key component to increase the ultimate appetizing value and the consumer acceptance. Why should we go for natural food colors? Because it is obtained from the plants, healthier alternatives to synthetic food colors. Also, it provides nutritional value to the foods, and it does not contain any common allergy-prone ingredients. The Future Market Insights in 2021 reported that the sales of natural food colors will pass the mark of $14 billion by 2028, and almost 75% of the consumers are willing to pay high money for natural products. Synthetic food colors, they are produced either by complete chemical synthesis or by chemical modification of several precursor compounds like coal tar or petroleum products. The common red synthetic pigments or natural food colorants available in the market are azorubine, allura red, amaranth, ponsiphora, and erythrocyan. These are already banned in some of the countries. And the most common ill effects of the synthetic food colors are thyroid cancer, hypersensitivity, that is sensory triggers and behaviors among the children, chromosomal damage, and neurodegenerative disorders. That is why there is a need for natural food color at the moment. Yes, you heard it right. We are all eating already crushed beetles. The deep crimson dye is extracted from the female coccineal insects, Dactylopus coccus. These Mesoamerican insects were formed, harvested, dried, and crushed for its bright coveted red color. Once a famous American company, on famous American calf chain, they use coccineal extract for its famous popular beverage, strawberry frappuccino. It contains the extracts from this coccineal insects. As it causes many allergic to humans, they already stopped using it. The common allergenicity in humans caused by these coccineal insects are allergies in children and patients, spasmodic cough, rhinitis, and breathing troubles like asthma if it is consumed on daily basis, and dyspnea and cracked and bleeding lips if the cosmetic product contains extract from this coccineal insects. So, what is the statement of problem here is, there is a market pressure for the need of natural food colors. Consumer prefers natural food colors over synthetic food colors, and so the manufacturers are also forced to find an alternative for synthetic food colors. Ultra niche crop, high returns from smaller area, that is Russell. The vernacular names of Russell are red sorrel, Jamaica sorrel, Guinea sorrel, Queensland jelly, and Florida cranberry. The crop is grown in northern states and northeastern states of India in small scale or in backyards. It is locally called as lumbadi or lumbari. The scientific name of Russell is Hibiscus subdarifa. It belongs to the family Malvaceae. The origin of the crop is West Africa. The crop grows well in all tropical and subtropical conditions. The crop can come up well in fertile and sandy loam soils. The crop is annual, bushy, herbaceous shrub that grows up to 8 feet in height. The plant was introduced to vegetable from Africa to Asia as early as in the 17th century. The Russell is cultivated for its bright red calices. The Russell major cultivating countries in the world are China, Thailand, Sudan, Nigeria, Mexico, Tanzania, Egypt, Senegal, Mali, Singapore, and Malaysia. Among these countries, China and Thailand are the major producers and competitors in the Russell market, and Sudan is known for its best calices in the market. This picture delineates the stages of calyx formation. The first one, the bud is in smaller size, then it grows into fully matured bud, then the flower blooms. The petals of this flower is caducous in nature, that it will fall off on the next day. Then the calices begin to swell. From the flower to the fully mature calices, it will take 20 days to develop into the fully matured one. At this stage, we have to harvest for the producing natural food color. The significance of Russell are, it is a potential good source of antioxidants, that is good for healthy skin, anthocyanins, ascorbic acid, dietary fibers, vitamins, and minerals. Russell is known for its brilliant red color and unique flavor, so it is suitable for producing the natural food color. It is also used to prepare a variety of foods such as jam, jelly, chutney, squash, and as coloring agents in ice creams, cakes, and beverages. First, the fresh calices are harvested. This is the structural flow of food color development. First, the fresh calices are harvested, and then it is dried. Then it is pulverized into powder. Then the aqueous or solvent extracts are prepared. If it is a solvent extract, the solvent is removed using rotavap evaporator. Then the Russell extract is mixed with the wall materials or stabilizers. Then it is freeze-dried to get the final output food color. Products from Russell. These are the products, value-added products that can be developed from Russell. Russell tea, wine, sliced cakes, jellies, tablets, candies, jams, etc. Already the Russell tea is very famous in the international market. The benefits of Russell are, it aids digestion, relieves the menstrual pain, anti-inflammatory property, prevents colorectal cancer, weight loss, antidepressant. It also has anti-cancer property importantly, and it manages the blood pressure, healthy pregnancy as it contains a high iron content, and moreover it tastes great. The market data forecast in 2021 reported that the global Russell market size is expected to reach $244.9 million by 2027, rising at a market growth of 10.4% compound annual growth rate during the forecast period. A famous Israeli company, Phytolon, biotech food color and startup, they raised $14.5 million to process its technology and commercialize the natural food color. From my work, I summarized that Russell, the little red rubies, can provide industrially viable quantities of red color. The lesser known crop can be utilized for its food dye, cosmetic industry to fabric dye production, and several value-added products. It also generates entrepreneurship opportunities and upliftment of the socio-economic condition of the farming community. Thank you for the opportunity.
Thank you. The next on the B line is about monolaurin. It is an advanced monolaurin she'll be talking about. It's an incremental innovation. And now the emulsifier is used in various decoration and all that. Structural properties are to be studied here. It's a creamy dressing, soapy, and it's a hybrid of oil and water combo. And this is being touched by Adira in her research. She belongs to Department of Processing and Food Engineering. She'll be talking on advanced monolaurin formulation from coconut oil as emulsifier for food industries. Now you can share your thoughts here, Adira.
Good morning all, I am Adira from second year M.Tech from the Department of Food Process Engineering, and my chairman Dr. M. Balakrishnan, Professor and Head Department of Food Process Engineering, and I am going to introduce my research, Advanced Monolaurin Formulation from the Coconut as an Emulsifier for Food Industries. The current scenario, the need of the emulsifier is increasing according to the development in the emulsion-based food product. Now, people are shifting the consumption pattern from the unprocessed food to the processed and ultra-processed food. And there is a decline in the trend of cooking and increase in the snacking pattern. And now people are more convenient. People are looking for the more convenient foods, and there is a huge market for the emulsifiers. The emulsion market size and forecast, the market growth will be accelerated at a CAGR of 5.39%, and in 2020 the market size is US dollar 3.1 billion, and it is expected to be US dollar 4.81 billion in 2028. There is an increase in the market size of food emulsifiers. And the emulsifiers is used to mix the two immersible solvents and producing a stable emulsion, and can be used in various food products. And there are different types of emulsifier. In natural emulsifier, there are plant-based and animal-based. In plant-based, some of the examples are gum acacia, carrageen, and pectin and starch. In plant-based, it is gelatin and lecithin. I am going to extract the monolaurin from coconut oil. Monolaurin is a monoacyl glyceride of lauric acid, which can be used as an emulsifier for food industries, and it is having antimicrobial properties. Why we are going for a monolaurin? The significance of monolaurin: it can boost the immune system and effective against the broad spectrum of microbes, and it increase the high density lipoprotein and regulate the increase in the insulin level. Hence, it's prevent the diabetes and also healthy balance of intestinal microflora. So although it's having these properties and its use is still limited in the food industries, the extraction method of monolaurin, they are going for an enzymatic method and chemical method. In chemical method, they are using p-tholin, sulfonic acid, sodium hydroxide, and potassium hydroxide. In enzymatic method, they are using Candida rugosa lipase, Novozyme 435, and rhizomucor lipase. The challenges: when we are going for a chemical method, it is done under high temperatures, so there will be a degradation of nutrient compounds and the purity of the final product is less. When we are going for an enzymatic method, enzymes are costlier and accessibility is less. So we have an alternative. We are going to develop a process protocol with the synthesis of lipase from the carica papaya lipase. First I collected the papaya lactose and synthesized the carica papaya lipase from the papaya lactose. So the extraction and encapsulation method: first I synthesize the carica papaya lipase from the papaya lactose, then I am going for enzymatic glycerolysis of the coconut oil, purification, then encapsulation of the monolaurin powders by spray drying method. Finally, we will get the powdered monolaurin, which can be incorporated into various food products. The scope and utility: it may replace the many other additives because it's having multiple properties. It can be used as an emulsifier, it can boost immune system, it's having antimicrobial properties, it helps to preserve the food materials, and it is non-toxic and stable and less cost. This can be incorporated into the food product, and this can be beneficial to the market by
incorporating in this emulsifier in the food and helps to enhance the therapeutic and functional properties of the food product. Thank you.
Thank you, Adira. Third presenter is also Yogeshwari. Okay, so both of you can come and stand here and you have to receive the questions. Audience, time to ask questions now.
It was a nice presentation. I want to ask on anthocyanin. Can you please tell me how photosensitive is anthocyanin? And the next question is whether it can be extracted only by solvent or water.
Okay, anthocyanins are surely it is light sensitive. We can't keep in the open for a long time. The anthocyanin will start degrading. Aqueous extraction also can be tried, like hot water extraction, cold water extraction can also be tried.
Then why you are choosing for solvents?
Solvent extraction efficiency is higher than aqueous, based on my study.
Okay, thank you.
So I have a question that will complement hers. Is anthocyanin hydrophobic or hydrophilic?
Hydrophilic, sir.
Huh?
Hydrophilic.
Then why do you go for organic solvent extraction?
The efficiency is more, so the color intensity is high in a solvent extraction.
No, please try to understand the philicity of biological molecules.
Okay, sir.
Whichever is hydrophilic, it can be extracted with water.
Okay, sir.
Whichever is not soluble in water, you have to go for
Solvents.
My question now is anthocyanin water soluble or water insoluble?
Water soluble, sir.
And what is the solvent you have used?
I have attempted in ethanol with HCl 0.1 ratios, 85:15.
Okay. So the next question is for you. So how your, what is the name of the compound? Monolaurin.
Monolaurin.
Right? Can you relate diabetes and monolaurin? You said that it increases insulin levels, right?
Sorry, it's regulate the insulin level.
But in your slide, it was the other way around. Please cross-check your slide, okay?
I will check, sir.
Hi, I have a question about the anthocyanin extract. Is it heat sensitive?
Sir?
Is it heat sensitive?
Yes, sir.
Maybe I like to put it more appropriately. If you use it in other food products which has to undergo heating process,
Yes, sir.
would the color be stable or will it go like when?
It will degrade, start to degrade when it reaches 100 degrees Celsius, sir.
Okay, thank you.
I just have an observation to both of you. See, when you take a research project, the research project should be in a manner that nobody else can replicate very quickly, right? I know that it's academic interest only, but if you have an interest to commercialize what you're doing, it should definitely be well thought through and ensure that you take slightly complex, not too complex problem where you can't complete your research, slightly one level above in terms of keeping the research component intact so that it can also give scope for extending into a PhD, right? And even a postdoctoral fellow. So I think it will be very nice that you take a holistic approach to be able to take a deep subject but then take incrementally. Okay, good luck.
Anyone else in the audience? Taken for granted you don't have any questions. We'll go for theme 5. Thank you, both of you.
Well, students, next you are going to have a very interesting presentation from Mr. Sachin, and he has come all the way from Hyderabad, and just to address you and also to enlighten you. Yeah. And the topic he is going to talk, and I can say two important areas he is going to touch upon. The first one is hydroponics. And probably if you could just, you can take your mobile and Google Simply Fresh, SIMPLY, Simply Fresh. Then you can see the amount of work he has done on hydroponics. So Simply Fresh is the biggest hydroponics production unit in Hyderabad. And he is the founder of that particular company, Simply Fresh. And I had the opportunity of visiting his farm. I think whatever we teach, and I think you can see everything in a mega stage. So basically he's an IT professional. So he did his IT technology in Australia and turned into a big entrepreneur and doing a great job in hydroponics. And because that's what it happens, convergence. And we agro technologists, we know about the crop, we know how to cultivate, we talk about season, we talk about pests, we talk about disease, and we talk about climate change, and we talk about this is the temperature the crop comes up. And for him, this is not much important because he controls everything. And through his hydroponics farm, he controls all the parameters from temperature to relative humidity to everything he controls. And he can create a microclimate as per his requirement. There his IT knowledge comes in rescue. So that is very important. His entire IT knowledge of programming. So based on the requirement of the crop, because I think when I went, I think he was cultivating many greens, and from greens to ashwagandha to turmeric he cultivates inside that farm. And so I think we are trying to download that YouTube and trying to show it to you. Or else when you have got time, just Simply Fresh, and you can see that how big that particular facility, SIMPLY, Simply Fresh. That's all. So then that's the first part. He is going to talk about that hydroponics, and things have changed a lot in hydroponics. You should be strong in agro, and as well as it's all about programming and what temperature, because you can change the temperature, relative humidity, irrigation pattern, fertigation, everything has been programmed. There is no need for any human intervention, only that programming part. It's against a need-based fertigation, based on the crop life cycle, because a crop at the early life cycle requires less nutrient, and during later life cycle or reproductive stage it needs more nutrient. So all these things are programmed, and based on the program the crop is actually irrigated or fertigation is done. This is one advantage. And the second one is the yield. The yield is multi-time, it's more. So that is the first part he is going to talk, and the second part will be on, he is also involved in using phytomolecules, or I can call this herbal bioactives, because today morning a couple of students talked about herbal bioactives, even just before people talk about the biopigments, and he is also into that particular sector. And again, just if you Google Zero Harm, and that company, he is producing lot of products for anti-diabetic, anti-cancer, and so many products he is producing using herbal molecules. And so that is a right person we have actually requested to come and discuss with you. The whole idea is any innovative thoughts or any innovative products or concepts we have, and probably I think he is ready to help us whatever the way I think that particular concept can be given with financial support, or the particular product can be taken into the next level. So that is the whole idea of this one. So now I think I request Mr. Sachin to come on to the stage. And this particular session is all about herbal nutraceuticals and animal care. This is a title of this particular session, herbal nutraceuticals and animal care. In this particular session, Mr. Sachin will be giving the lead talk. Then 4 students, PhD students, will be presenting their research to the August gathering. So that is what I think the program for the next half an hour or 45 minutes. And so now I request all the 4 presenters to come onto the stage. And yeah, Sachin is setting things ready. No problem. So I think you can come onto the stage. I'll set it right, Sachin. Come. So give a big round of applause to all the students and the speakers. Please, okay. So may I request Dr. Prabhu to do honors for the chief guest with a bouquet and a memento. The speaker, Mr. Sachin. And more or less it's set, but now I request, because Sachin, you can give a extempo introduction about you, and I can just, he sent his one-page profile, but I think it looks very impressive. I think I need more time to go through, but quickly I think he's the founder and director of Zero Harm Sciences, and 21 years of experience in agri-tech and information technology. And he is the previously founder and CEO of Simply Fresh, India's largest state-of-the-art precision farm hydroponic unit. And he has a strong background in information technology with a strong understanding of agricultural technology and farming technique. An IT professional pursuing a lucrative career overseas returns to India to start India's largest hydroponic precision farming company, Simply Fresh Private Limited. And the story is nothing less than an inspiration to the young generation of this nation who are looking at agriculture and ag tech as their preferred career choice. So you can't have a better person than Sachin to motivate you. Having worked in Australia, he holds an enriching background in information technology with a strong understanding of agricultural technology and farming technique. Simply Fresh was set up with the motto of provide safe, hydroponically grown, pesticide-free, traceable, fresh produce and medicinal plants with high nutritional value and active ingredients level. Simply Fresh uses high sustainable precision farming techniques to grow all plants in all AI-controlled automated greenhouse. So it is an AI-controlled automated greenhouse where an ideal environment is created for plants to grow. So I think he has developed a proprietary software, Farm in a Box. So this software controls the entire ecosystem. It creates a microclimate. So it is a proprietary, it's a IP-owned Farm in a Box software. So in recent years, there has been a tremendous shift in health consciousness and how people started talking health in more serious way, because after COVID everyone is looking for about healthy products, and as well as people are talking much about our native wisdom. And for information that during the COVID and post-COVID, one herb was sold highest in Amazon in US. That is nothing but Indian turmeric. Indian turmeric was the highest sales in Amazon after COVID. So now we are going back to our native wisdom, and Sachin is having another company called as Zero Harm Sciences, and the whole idea is developing or giving these bioactives in a more enhanced bioavailability. So with this small background, I think I invite Mr. Sachin to present here his ideas about how that science or students can be linked with industries so that the commercialization happens smoothly. Mr. Sachin, please.
Thank you, sir, for the introduction. I will just play with the I will just take you through the site which we have built in Hyderabad, and we'll just go through it for a few minutes, and then we'll dig into the presentation. Perfect. So what you have seen is India's largest hydroponic setup. Why it was created was there was a need to have a very consistent product when you are doing extraction or when you are doing any bioactive or for that matter any phytochemicals. So the consistency was a big issue. So how do you overcome it? One is definitely the tissue culture, which we have seen in previous presentation, which gives you a very consistent mother stock. And then the growing conditions and also the nutrients. What the plants eat is what the plant synthesizes and deposits it into alkaloids or other aspects which we are interested. So that's where the Simply Fresh came in. We control the nutrients, and it's all automated because it's quite challenging to manage close to 16 different nutrients on live basis. And plants are no different. They are just like human beings. They have different requirements in summer. They have different requirements in winters. They have different requirement when it is humid. It has different requirement when it is very sunny. So what we do is we trial a plant in a lab, and we understand its nutritional requirement, climatic requirement. We understand how it behaves differently under different stress situations. And we replicate that recipe on a live basis. So the nutrients are managed pretty much live, and that's where the Simply Fresh grows medicinal plants and fresh produce. So these are some of the plants which we grow for extraction purpose. Then we started another company called Zero Harm, which is almost 2 years old company. So idea with Zero Harm was how do we use more aqueous extractions? How do we make the issue with nutraceutical is the bioavailability, because a lot of these are extracted using solvent, so they are not water soluble. The moment they are not water soluble, the absorption is very less in the body. So we tried and we applied couple of patents to use water under high pressure, high temperature with different combinations to extract, and that's where Zero Harm Sciences was born. So right now, based on my understanding and the exposure, we feel there is a new industry getting formed, which is a combination of pharma and nutra. So why there are a lot of issues with the pharma as such? Pharma is more when you have a problem, you go and talk to a doctor and take a medicine. But whereas nutra supplements has been more as a preventive approach. A lot of people have started taking supplements, vitamins, pills, more as a preventive. Like I have seen in US, a lot of people are taking 5 to 16 pills a day. These are all very preventive style. And people are very much worried about their health. They don't want to fall sick because healthcare is very expensive in a lot of Western countries. And the healthcare is not that easily available also. So people are now thinking that I don't want to fall sick. They are not thinking about what should I do when I'm sick. They're trying to be more proactive. And that's where all of this have started coming in. But the inherent problem is poor bioavailability. Second is slow response rate. A lot of nutraceutical are consumed over the time and they take long time to start showing the effect. So with today's generation, everybody wants everything to be instant. They don't have patience to wait for 6 months, take some pill and wait for the results to come after 6 months. And high raw material cost compared to pharma, nutraceutical extracts are very expensive. There are no side effects available over the counter, which is again a big issue. It is not regulated as medicine. It is still evolving. But the phytopharma, we are trying to use nanotechnology to increase the bioavailability of these supplements. Low cost, because once you convert it into nano, the dosage comes down drastically because of the surface area getting increased and the absorption gets high. And the preventive and cure, no side effects because the dosage has been reduced. That means the toxicity chances are low. Inherently, Ayurveda and a lot of Nutra products have toxicity issues. So, phytopharma has been coined, the word has been going around for a long time, where you are purifying nutraceutical medications or nutraceutical extract to a pharma grade. So, these are some comparisons how this industry looks like. So this is the platform we have developed at Zero Harm. The platform is basically using water wherever it is possible to extract rather than solvents. So we are trying to reduce the usage of solvent as much as possible, and we are picking one extract at a time and trying to use whether we can do it with certain technologies which have been patented to do water-based extraction. Second, we are trying to use nanotechnology, breaking the particles into smaller size. So the whole purpose is to, one, make it pure, two, make the dosage lower and lower as much as possible. Third, encapsulation, because a lot of water, once you use water, the nutrients are available in a burst mechanism. But you want a very slow release because you want body to take its own time to absorb. You don't want to just burst it and forget it. So you need encapsulation. So encapsulation enables us to have very sustained release. So it gives us more time. It gives us more ability for body to absorb. Then we can put a charge on it. We can put a positive or a negative charge on the molecule, which again enables it to be a very targeted delivery. Like COVID is a positively charged virus, and if curcumin is negatively charged, it can go and attract and join itself. So nutraceutical market is pretty much broken. Sorry. So it is broken into two spaces today. One is functional food and beverages. Second is dietary supplement. And as you have seen in previous presentation, this sector is growing really big. And the growth is there. There are a lot of new companies are coming in. A lot of innovation is happening. And it's a space where I would encourage a lot of innovation to happen. I would like a lot of students to look at this space very seriously. How they can use innovative technologies, how they can use genetics to make good planting material where the alkaloids percentage is higher. For example, if can we make a turmeric plant with 11% curcumin consistently? Can we reduce the oil in turmeric and increase the extractability of curcumin? So there is a lot of space and there is a lot of demand, as you can see in the color space, supplement space. There is huge demand for plant-based multivitamins and minerals because most of the multivitamins and minerals today come from China, and there is a good demand if we can make it plant-based. Can we make multivitamins and minerals using plants? So these are some of the companies which are into this space, and they have been given extremely high valuation. Lot of money has been raised, and there is a lot of work happening outside of India also. It's not just that nutraceutical has been restricted to India anymore, and a lot of companies in US, Canada, Switzerland, they are looking at this space very closely. More because of hydroponics, more because of the way you can grow any plant, irrespective of the origin of that plant, because you can create the microclimate, you can containerize it, you can grow centella in a container rather than growing it in wild jungle, because the technology today is enabling you to grow anything anywhere. This is just the market size, as we all know and as we all have seen. So what are the challenges in this space? First challenge, it's a scattered market due to various regions are growing it. A lot of plants are coming from Madagascar to Amazon to India to China. There is no control as such who is growing what, and it's open for anybody to grow anything. So even the batches which you get from the same field is so different that the extraction becomes a lot more challenging. It's not consistent at all. Lack of standardization. Right now, this space is not governed by FDA. It doesn't come under FDA. So there is lack of standardization. A lot of room is there to standardize these extracts and come up with certain certifications. Purity, again, as I said, it's a plant naturally grown, and every extract will be different. How do you purify it? How do you standardize it? And how do you set a benchmark in the industry? Like extract of ashwagandha will be XYZ. Who will certify it? Who will approve it? Who will make sure that every ashwagandha is saying, where is that SOP? That's the space today. Nobody has answers. Everybody sells ashwagandha. They call it ashwagandha with certain ethanoid content in it. But what about the rest of the material in it? What is it? What is that the remaining 90% is? That kind of detail is not there today. Lack of clinical trials. Lot of tall claims are there in this space that the diabetes can be reversed, your liver, fatty liver can go away, but there are no detailed clinical trials happening. And there are a lot of companies who are now trying to get into it to secure a claim so they can put it on their product, saying that my product has went through clinical trials. So that's the space which is happening. Poor bioavailability, dosage, we just touched the topic. Future, I think phytopharma grade of plant-based molecules, I feel, is the future. People are looking for very pure material. So they can standardize the tablets, they can standardize the dosage, they can standardize the usage of it. Regulations, I think at some point government or some bodies will step in to regulate this space, because today it's everybody's market, anybody can sell anything. So there is no regulation as such. Awareness about preventive properties of this plants, there is very less awareness in the space. People are not very aware the wisdom what we had from Ayurveda, what kind of plants we have, what kind of extract we have, what are the preventive properties they have. And end-to-end traceability is missing. Right from the field to tablet, there is no traceability. You don't have that kind of traceability today. These are some of our products which we have created using this technology. This is Oncolis, which is targeted towards cancer, diabetes, arthritis, iris for the eyes, and again this has an extract of marigold flower and paprika. Curcumin, this is, we call it holistic curcumin because there is a lot of goodness in turmeric, not only just the curcumin. So we extract other parts of turmeric also, convert that into nano and make it 100% water soluble, and the absorption is extremely high. So this is what goes into a holistic curcumin tablet. This is a probiotic. It's a combination of 24 unique strains. Ashwagandha, again as I mentioned, ashwagandha has a lot of other goodness other than withanolides. So we extract all of that using water. We convert it into nano, we encapsulate it, and then we put a charge, and that's our product. So today our company is marketing these products in 5 countries. Calcium, again milk-based calcium, and calcium and curcumin is one of the combination which is very popular abroad. People have seen a lot of benefits. One, their bones are getting stronger. Second, their inflammation is coming down. So one tablet is giving them both the benefits. And ashwagandha has been used widely in US for sports. A lot of sports people who are into sports, workout, gym, or any kind of physical sport, they have started using ashwagandha. One, it is allowed by the authorities to use ashwagandha. Two, they are getting very good results. So we came up with a product which is calcium and ashwagandha. Vegan omega, again, which is a very innovative space. We are extracting omega from algae, not from the fish. So we breed the algae, we extract the oil, then we freeze dry it, convert it into powder and make them into tablets. So there is a scope where you guys can come up with some new algae or new type of it, which is a lot more odorless or which has much better palatability. That's the presentation from my side. Happy to take any questions.
Yes, students and friends. So now you know what is happening in the market. So when we talk about extraction, when we talk about bioavailability and so many things, so here's given with some case study. For example, what nanocalcium, because we talk about ashwagandha, I think agri students, we can talk a lot about ashwagandha, how to cultivate and all other things. But how to convert that into a industrial scale product and what is its current status, what is a global scenario, national scenario, what is the requirement of all these things. So now I think Mr. Sachin has given a good idea about that market condition. So now probably I think I request you just to come out with your queries before I think the student presentation will start. Questions, please. Madam. So because it's not question, madam, you are opening up the debate. That's very interesting.
Thank you. Sachin, very interesting presentation. I think we got two perspectives of what you're doing and then what you are actually, the future is also in terms of people going in this one. I just wanted to ask largely, how do you do these kind of research? Do you collaborate with university? And what are the kind of research opportunities exist for our students in TNAU? That is number one. Number two is that, are you funded? What's the plan for your company? What's your size? What's the growth plan?
So, first question, we are funded. We've been funded close to 250 crores. And growth plan is to take this in different countries and replicate these structures. Number two, I would love to collaborate with universities for innovation. There are a lot of problem spaces where we can jointly work and try to see if we can come up with some kind of solutions. But unfortunately, in commercial space, we are always pressed against time. And that's where we need to work very, very fast, because things move very fast in the industry. And yes, would love to collaborate on certain projects, certain problem spaces which has good commercial viability.
Just one, just a very, very fundamental question. I may be naive to ask this question. It's always said that any crop grown in a natural environment on the earth retains everything much better than this kind of a setup, right? So what is your take on that? Do you agree to that? What is your take on that? You should give me a frank view.
Yeah. So you cannot replace Mother Nature. That is 100% absolutely right. But for a commercial project, like for example, you take tomatoes, which comes out of hydroponic farm, are much, much better tasting, much better flavors, and shelf life is a lot more. Hardness is there, shine is there, because the plants, end of the day, work with microbes to communicate what kind of elements they need for their growth. So we are replacing that by saying you need iron now, you need zinc, you need this much of boron. All that we readily provide it. So we make the plant life a lot more easy. And it's a kid which is taken care from childhood in a very good locality, or in a hospital, you can call it. But it is definitely not as strong as a plant which is grown in soil. It's not hardy. You have to take a lot more care of them, because they are soft. But the end product, you can compare it. It has 12 nutritional elements, whereas the soil grown has 3. So the nutritional value for humans is a lot more when you consume a hydroponically grown produce. And for nutraceutical space, you can pretty much control the alkaloids which are getting synthesized in the plant. Because end of the day, the building blocks, you are providing them at the right time. So it's get better. But if you ask me, the taste-wise, I would prefer the farm-grown one, hydroponically one. And the field ones are not consistent enough. So that's where the difference is.
Any more queries from student side? I know that refreshments are being served, but you take that one without disturbing the speaker. Okay, right.
Students maintain silence, no? It's an intellectual gathering, no? It's not a fish market. Understand?
Hello, sir. Good morning, sir. My name is Rakshit. This question is regarding hydroponics, sir. You tried in hydroponics, you are doing in horizontal farming, right, sir? have you ever try this vertical farming? Because in microclimate area, the area will be less, means if you're doing in horizontal, means if you're doing in vertical means you can harvest more area, so surface. Have you ever tried that vertical farming in your microclimate area for hydroponics, sir?
Yeah, it's a very good question. And the answer is both of the technologies are suitable. So I always believe it's not about high tech or low tech, it's about the right technology. What is required for your country, what is required for your region, what makes economical sense. In India, land is freely available, meaning it's abundant. We have plenty of land. There is no shortage of land. So when you go vertical, you have a problem with light. Creating artificial light is very expensive. Number two, you have to create an artificial atmosphere also. So right now, the kind of price you get for your end product doesn't justify vertical farming in India at this moment. So maybe in future, when things get a bit expensive, when people start paying a little bit more for their vegetables or the raw material, then vertical farming will make sense. But at the moment, vertical farming is very expensive to grow.
Okay, sir. Thank you, sir.
Good morning, sir. Any idea about multivitamin or nutritional tablets? Because you are doing nano formulations for calcium and all. Is there any possibility to do that multivitamin thing?
We are releasing multivitamins which are extracted from plants, vegetables and superfood. So that is coming into the market by end of this month. So there is very good scope and there is very good demand in business to business also. There are a lot of companies who are looking for that type of raw material which is totally derived from plants. But the testing is very big challenge. How do you test it? How do you standardize it? And how do you give consistent batch? But yes, we have a product with that.
Sir, the question is, you said the testing will be difficult. So we have normal chemical multivitamin tablets. And I guess the procedure will be almost same to test them. What makes the difference between chemical and plant-based?
So when you extract from plant, it comes out as a composite. It doesn't come independent iron. So if you extract it from guava leaf, a lot of amino acids and other things come out of it. So to standardize it and test it, what other things are there gets a little bit challenging. You can test that it has X number of iron in it, but you cannot test what all other things it has.
Thank you.
Keeping oneself beauty is not Mona Lisa's property. It's also not Isabella's ownership. Now there is a lot of countenance on deterioration of the face, skin, exfoliation, cleanser. Lot of beauty parlors are up and coming and all that. Heels, acne, reverse aging, everyone without gender. Lot of naturals are coming in. So what's new will be revealed by P. Divya Bharati in a product innovation belonging to Department of Nanoscience and Technology to talk on antioxidant nano formulation from orange peel. You'll be given 10 minutes to complete. Yes.
Hello all. I am Divya Bharati from Department of Nanoscience and Technology and I am doing my third PhD. So here I am going to talk about my innovation on developing antioxidant nanoformulation from an orange peel. So in this presentation, I would like to talk about what all the creative solutions we can produce from an orange peel. So start with this presentation. It's a global scenario of orange production. So as per the report, the 2020 to 2021, the orange production was about 48.57 million metric tons. So the right side of the slide indicates what are the production versus the processing of the orange. The orange is mainly used for the production of juice. So among 48.57 million metric tons of orange, 31 million metric tons or citrus fruits are processed globally for juice industries. So in which 15.6 million tons of citrus peels are generated per year. So here comes the scope of the orange peel. So as you can see in the slide, the orange peel has 3 layers, which has flavido, albedo and the oil glands. So you can see that flavido layer has a lot amount of carotenoids, flavonoids and the chlorophylls, and the albedo layers made up of sugars like pectin, cellulose and hemicellulose. So then in oil glands it's mainly made up of an essential oils. So I could say that citrus byproduct resulting from the citrus manufacturing industry, it's account for about 50% of the total fruit weight. So citrus industry byproducts or especially the peels, which have been found to be major source of phenolic compounds, especially limonene, an important bioactive present in the orange peel. So we can extract this natural antioxidant from the orange peel and we can convert it into an beneficial nano formulation for human consumption. So starting from citrus waste, the primary product we can develop is an extraction of limonene, pectin and cellulose from the orange peel waste. Followed by the high value molecules such as biodegradable polymers. That is cellulose, hemicellulose, pectin. These are all the second layer we can extract from the orange peel. So the third one is we can extract even carbon nanodose and the porous carbon from the orange peel waste. Finally, 3D printed objects also can fabrics can also produce from the orange peel waste. So I could say that it is a wealth from waste concept. Here comes the hypothesis of the invention. This innovation comes under the category of substantial. We have developed an innovative and integrated extraction technique using a important solvent called deep eutectic solvent with an innovative extraction technique of ultrasonic method. We have extracted the important components from the orange peel. For that, I have prepared an innovative solvent like deep eutectic solvent using an hydrogen bond acceptor and the hydrogen bond donor. So what is difference between the aqueous extract and the deep eutectic solvent based extract means if you are using an aqueous method of extraction, you can extract only the hydrophilic molecules from the plant material or the bioactive. So if you are using a deep eutectic solvent, you can extract efficiently both hydrophilic and lipophilic moieties from the plant materials. So nowadays solvents are playing a major role for extracting an lipophilic components from the bioactive material. But in case of this project, I have used deep eutectic solvent to extract efficiently essential oils, pectin, biominerals and polyphenols from the orange peel. So I have not only have extracted the antioxidant or polyphenol. Initially, I have extracted the oil from the orange peel waste. After that, I have used a deep eutectic solvent and ultrasonication method to extract the biominerals and the polyphenols, even pectin gel from the orange peel. So way forward. So I have extracted the compound. What next is the encapsulation of this extract. So the bioactives are further encapsulated with nanoKS with a nano encapsulation technique to improve the efficiency or the therapeutic value of the orange peel. So these are all the way forward of our research. So we need to optimize the nano encapsulation technique followed by the determination of encapsulation efficiency, and the bioavailability study has to develop to further develop nanoformulation. So this is the utility for the industry. Mainly oranges, not only peels, it has seed, peels and pomace. These are all the 3 possible ways we could, has a large spectrum of useful for the industry in following by food sector, packaging sector and animal feed, pharma sector, cosmetics and biofuels. So what is the impact of this innovation was, we have used a non-conventional extraction method with a green solvent. I could say there is a green deep eutectic solvent and the multiple product from the single fruit waste and potential antioxidant for nanoformulation from an orange peel waste. Thank you.
Thank you, Divya Bharati. Now, the prevention is better than cure. Everyone is longing and yearning for safe, healthy, affordable, and optimal health and quality diet and all that. For that, nutraceuticals comes in a big way to rescue this, and we are going back to traditional rice too. Now we have got P. Mokita of Department of Nanoscience and Technology, who will be talking on the nutraceutical nano delivery system to enhance the bioavailability and the therapeutic potential of traditional Kavni rice. Now the stage is you to share for 10 minutes.
Hi, good morning everyone. I am Mohita and I am doing my second PhD in Department of Nanoscience and Technology. And my innovation is about nutraceutical nano delivery system to enhance bioavailability and therapeutic potential of Kavni rice. As you all know, rice is the major staple food for Southeast Asian people, which feeds more than 500,000 people in our country. And there are more than 1,20,000 rice varieties available in our country. But increased white rice consumption always leads to major threatening disease. And one among them is diabetes. As India is the global capital of diabetes, with approximately 96 million people are affected by diabetes. And also, the pre-diabetic people are in danger for developing insulin-resistant diabetes. Have you ever think about what is in your rice? I think maybe few, but everyone should think about what is in your rice. Why? Because whenever we consume white rice, it lacks multi-nutrients, vitamins and minerals. And to overcome this, and also the glycemic index, which plays a major role in causing diabetics. What is glycemic index? That is the term which indicates the conversion of starch into blood glucose level. Whenever we consume white rice, the glycemic index will always be higher. That is, it will be higher in the range of more than 70. So we need to, so we are in the urge to have low glycemic index food varieties. For prevent diabetes. So in order to prevent diabetes, there are a lot of medicinal or therapeutic rice varieties in our country. And over 2,000 traditional land races of rice varieties are available in India. But why one among them is Kavni? Why I have chosen this particular Kavni rice is because this particular Kavni rice having a very low glycemic index, which is less than 50. So in this we identified and the major phytochemical which is responsible for anti-diabetic activity in Kavni rice is anthocyanins. So the anthocyanin where we have identified a total of 6 anthocyanin that is present in Kavni rice using liquid chromatography mass spectroscopy technique, and the predominant anthocyanin present in Kavni rice is cyanidin 3 glucoside, which is 91.13% of total anthocyanin, and the quantifiable amount is 27 mg per gram of Kavni rice. And the other predominant one is pionidin 3 glucoside, which is 4.74% of total anthocyanin. And the quantifiable amount is 2.9 mg per gram of Kavni rice. All these quantification can be done by using liquid chromatography mass spectroscopy technique. But the major disadvantages associated with this anthocyanin phytochemical is it is extremely unstable. Whenever we can consume this Kavni rice as such, only less than 1% of anthocyanin will be available in our human system. Why? Because whenever we intake any food, the gastrointestinal tract where our every food will get absorbed. But here, the gastrointestinal tract, the pH of the gastrointestinal tract is alkaline. So this particular anthocyanin will be stable at acidic pH, but it will be unstable at alkaline pH. So we need to, it is one of the major disadvantages. And another disadvantage is temperature sensitive. So whenever we can cook this particular Kavni rice in a pressure cooker or pressure cooker or rice cooker, there will be a greatest loss of anthocyanin. Only we can get 10% of anthocyanin which will be available in our body system. And also the rapid metabolism. So whenever we intake this Kavni rice, within an half an hour this anthocyanin will get metabolized and it will rapidly eliminated by urine and plasma. So to overcome this, we have a nanotechnological. So to overcome this, we have a nanotechnological approach to encapsulate these particular bioactives into a nano carrier system. So here I have been using natural protein and polysaccharide based nanocarriers for encapsulating my anthocyanin into a suitable nanocarrier. And I am in the process of developing a very good nanocomedy product by encapsulating this particular anthocyanin in a stable nanocarrier system. By using this nanotechnology, we can enhance the bioavailability of this particular bioactive, and also we can achieve slow and sustained release of phytochemical in our human system. And the way forward in my innovation, I am developing a stable nanoformulation which is ready to eat nanoformulation in the form of either liquids or either pellets or either gummies. And then we need to go for in vitro therapeutic efficiency of this antibiotic drug loaded nanoformulation. And further, preclinical and clinical study is very much important, and also the metabolic targets, how this particular anthocyanin will be acting in our human system to enhance the antidiabetic activity. So the scope of this nutraceutical industry, it is one of the rapid radical innovation where we can develop effective antidiabetic treatment in the near future. And it is also a multi-million dollar industry, and the day is not so far that we have a very good nano antidiabetic drug in the near future, and we can proudly say that our India is a diabetic free India by the year 2050. Thank you.
Thank you, Mokita. Let your vision and dream becomes true to make us diabetic free, because India is a diabetic capital in the world. And the third on the B line is Chitra, who will be talking about the health is wealth. When everything is lost, nothing is lost. When health is lost, everything is lost. Now what she is going to do on her nanobetaloins as health supplement, Chitra, belonging to Department of Nanoscience and Technology, Coimbatore, will be sharing ideas. It's a product innovation.
Good morning to one and all present here. I am Chitra, doing my second PhD in Nanoscience and Technology Department. I am going to present my innovation, that is nanobetaline as health supplement. So we all know beetroot, it's a common vegetable. So everyone can think, what's the special thing about this beetroot? So you can think that every day I am eating this beetroot. So what is the thing I am going to say it is about a health supplement? So let us see. So I can say beetroot as a remarkable vegetable. So nowadays, people are moving towards the word called functional foods. So the functional foods, they are the ingredients that offer the health benefits that extend beyond their nutritional values. So this beetroot is also considered as such a functional food with huge health benefits, because they contain the major source of bioactive compounds like betalains. The betalains, they are the major source of bioactive compounds in this beetroot, which gives the typical red and purple color to this beetroot. The color pigment responsible for the color of this beetroot is betaline, and also we can say this betaline as a medicinal pigment. So, and the next one is the dietary nitrates, the energy giving nitrates to the humans, and the phenolics, potential antioxidant compounds, vitamins like retinols are present in this beetroot, and minerals like potassium, magnesium, calcium, particularly iron, everything is present in this remarkable vegetable. So the major phytonutrients of beetroot are, like first one we can say split into two, like one is the biologically active phytonutrient and the another one is the other phytonutrients. In this bioactive phytonutrients, the major content is the betalains and the polyphenols, phenolic compounds, saponins, flavonoids, vitamins, nitrates, everything is present. Among them, I am going to detail about the betalains. These betalains are made up of two compounds. One is beta-cyanins and another one is beta-xanthins. So the beta-cyanins are the red color pigments, which is typically responsible for the purple color of the beetroots, and the beta-xanthin are the yellowish fluorescent color pigment. So this beta-cyanin we can say as an example betanin and isobetanin. For beta-xanthin we can say as indica-xanthin, vulga-xanthin, like that. And the other phytonutrients present in this beetroot are proteins, carbohydrates, amino acids, fatty acids, etc. This betaline is majorly derived from the aromatic amino acid tyrosine. So we can say it is an enormously rich of the aromatic amino acids. So these are the major phytonutrients contained in beetroots. So I come to betalines. So the betalines, these are the, we can say it is an unique pigment. They are the unique water soluble nitrogen containing vacuolar pigments. So they have exceptional medicinal properties. So we all know betalines as a coloring agents. Apart from that, they are having enormous nutritional and medicinal properties. So these betalines are in both industry and manufacturing level, they are limited up to as a food colorant only. Apart from that, these betalines nowadays only they are coming as an functional pigments and bioactive compounds. So this betaline, they can be used for these kind of functional properties. So I start with cardioprotective. So nowadays there is no age limit for the cardio diseases. So this cardio diseases, they can be due to some vasodilation like that. So this betalines, they can act as a potential antioxidant and anti-inflammatory agents by potentially scavenging the free radicals and provide as an anti-inflammatory agents, thus protecting our heart. They can also act as a blood purifier. We all know the role of hemoglobins attaching the oxygen to our red blood cells and carrying through our capillaries. So this betalines, they are having the enormous amount of nitrate contents. So this nitrate contents, they attaches to the hemoglobin and provides an additional carrying of the oxygen into our blood capillaries, so that they can also mimic as a vasodilator. So there is no risk for the heart diseases and all. And also it is an effective hepatoprotective and as an cerebrovascular protection also. We can say it can enhance the cognitive functions. And nowadays these betalines are used for the stamina increase. So in sports field, the athletes are nowadays using this betalines as their energy drinks. And also one of the major property of the betalines are they are reported as the chemoprotective agents. So for skin cancer and lung cancer, they reported that betalines have reduced the anti-tumor, they have reported as the anti-tumor agents, and also they risk the production of multi-tumor productions in the human bodies. So as I said, nowadays athletes, they need more stamina and powers during workouts, everything. So this betalines are having enormous amount of nitrate contents. So this nitrate is further when it is consumed, it is converted into nitrate and then into nitric oxide. So this nitric oxide, it combines with the hemoglobin of our blood and it efficiently carries the oxygen into our blood capillaries and it acts as a vasodilator. So these athletes, they can get more stamina and energy without any so what is my scope of innovation means? This betalines, they are having this much enormous amount of medicinal properties and nutritional values. So the major disadvantage is that these betalines are highly sensitive to light, pH, oxygen, everything. So whenever the pigment is, whenever we cut the beetroot and we can see our hands, they are purple and immediately it will be turning into brown. So that is due to the degradation, due to the enzymatic degradation, and also it is very heat sensitive and pH sensitive. So what happens means when we ingest the beetroot, our stomach pH, we all know the stomach pH is about 2 to 3.5. After that, the small intestine, it will be like 6.5 to 7, and the duodenum, it is about 7 to 8 pH. So the all along way, the betalines have to travel. So the way when it reaches the stomach itself, it gets degraded. So that needs a protection to that betalines. And also when it get degraded, the bioavailability is also reduced so much. When you consume about 500 ml of betalines, only about 0.3% of that dosage is absorbed into your body. For that reason, we should protect this betaline with the nanotechnology inventions like nano encapsulation and nano emulsions. For that reason, I have developed this nanotechnology interventions for protecting this betaline from degradations. So my innovation is a substantial innovation because there is already some works going on this particular field. I'm in particular developing a green solvent technology based innovation with ultrasound based technology. So basically like this novel extraction technologies are like ultrasound, microwave, pulsed electric field, membrane technologies are there for extraction. I'm particularly using this ultrasound assisted extraction technology for extracting betalines. And then I'm stabilizing the betalines using nano encapsulation technology and nano emulsion technology to provide them higher shelf life and sustainable release in our body for enhanced delivery system into the humans. So this is the current status of my innovation. What I did means, first I extracted the betalines with ultrasound assisted technology. This is the crude betaline extract. So with betaline, all other flavonoids, saponins, carotenoids, everything will be there. But the major content of about 85% is betalines. And then I am concentrating the betaline, like only the betaline I am concentrating by stabilizing the pH. And then I am converting them into the nano betaline, that is the betaline nano emulsion. This is the nano emulsion betaline I have developed. That is the coarse emulsion I have viewed under the microscope before converting into the nano emulsion. And also I have dried the betalines and converted into the betaline, nano betaline powders. So it is completely water soluble. You can dissolve it in any water, milk or any substances and we can consume. Since it is completely water soluble, it is fully bioavailable to your body, and also we have encapsulated that substance, betaline substance. So it is highly protected and it won't degrade under the harsh substances like stomach pH and small intestine and then duodenum and all. So these studies I should do, like I have to do the stability study. It's currently under ongoing process and I am in the trial and error method. So at present I have developed the nano emulsion and nano encapsulated betalines with some suitable wall materials. Again I have to choose some suitable nano material carriers for the efficient delivery of bioactives betalines and also for the efficient encapsulation efficiency, and also I have to study the bioavailability studies because it is an important parameter to determine the efficiency of the, my nano betaline. So what is my major impact on my innovation is that, so first one, I extracted completely betaline with water. There is no other any preservatives or any harmful chemicals or anything. So it's completely green solvent technology, and I have stabilized using the nanotechnology. Mine is a completely nano betaline, which is in a nano range, so that will be a complete absorption into the body. With this nano betaline, we can fabricate this product into any edible products. There is no restriction for this. We can incorporate into any edible products, and also there is the chance for easy upscaling in the industries. It can be used in like drug industries, medicinal industries, sports industries, everything. Thank you.
Thank you, Chitra, for an exhaustive presentation. Now, we have got a lot of compounds on them, bioactive plants, terpenoids, alkaloids and phenolic and all that. We are very curious to know what kind of nano antibiotics she had extracted to countenance the Staphylococcus aureus, which is most dreadful in poultry industry, kept at stake. L. Shivashankari of Department of Nanoscience Technology will be here to talk on nano antibiotics from plant bioactives for the control of multi-drug resistant Staphylococcus aureus in poultry birds. Shivashankari, it's your time.
Good morning, everyone. I am Shivashankari, doing my second PhD in Department of Nanoscience and Technology, and guided by Dr. A. Lakshmanan sir, Dean SPGS. So I'm here to present my innovation, nano antibiotics for ensuring organic meat and egg. So before moving on to the actual presentation, we would have read in our smaller classes, Cori, Mayu, therium அப்படினு we would have heard, right? So which says the country higgs would wander around in fields and it pick up bright herbs or worms that is required for them by themselves, and they were still healthy, right? But today, where the chickens are grown in poultry farms, like they are dumped like 1,000 and 2,000 birds under a roof, this is not the case, right? So more insights can be found through listening to a conversation between a country chick and a poultry chick.
Hey Broily, you seem to be down in the dumps. What happened?
Yes, dear Lucky, I'm done with the amount of antibiotics I've been fed every day.
But it's for good. They protect you from infections. My human dad gave me some herbs too for the same purpose.
Maybe, but the microbes infect me even more severely after taking more antibiotics. Do you have any idea why it's happening?
Hope you're not aware of superbugs.
No, what does that mean?
When a single pathogenic organism is resistant to more than one antibiotic, it is said to be multi-drug resistance. Pathogens with such developed resistance are superbugs.
Ooh, that's frightening. Can you add more to it?
I've just heard about it from a human friend. She can explain more about it to you and to the audience. Hey Shiva, will you do it?
Yes, Lucky, I can do it for you. So first, let's know what is antibiotics. So the antibiotics is used to disrupt the essential processes or structures in the bacterial cell. They could either kill the bacterium or they could slow down the growth. Depending on this activity, they can either be classified as bactericidal or bacteristatic, right? So this definition clearly shows that antibiotic is a great gift to human race, right? But still, I doubt, because of this antibiotic resistance. The pathogens are very smart than humans, that they have already started developing resistance against any antibiotics we give them. So what is the mechanism involved in that? Various mechanisms that includes a flex pump, where the pathogens pushes out any antibiotics given to them, or they block the penetration itself. If the antibiotics never penetrates into a bacterial cell, then the antibiotic activity is not possible. The third mechanism involves the inactivation of enzymes that are responsible for antibiotic activity. The third one is target modification. As I mentioned, they're very smarter than us, because we design antibodies based on the antigens, right? And if they completely modify the targets, then it is not possible for the antibiotic we give to be effective. So these are the various mechanisms the pathogens use to create an antibiotic resistance. So chicken. We all love chicken, right? The term "I love chicken" is very common nowadays. The chicken on plate, not a living chicken. So what does this chicken contain? 165 calories, 31 grams of protein, 3.6 grams of fat. Everything in 100 grams of chicken we feed. So it seems to be a very proteinaceous, healthy diet, right? But still I doubt, because it also contains n number of antibiotics like oxytetracycline, chlorotetracycline, doxycycline, enrofloxacin, ciprofloxacin, neomycin, and still many more antibiotics in it. A study is also conducted in India in 2018, and it says in the important muscles like liver, kidney and breast muscles in chicken contains 44% of antibiotic residues in it. That includes all these antibiotics. So out of it, only 17% as a antibiotic residues of one antibiotic, all other as a multiple antibiotic residues in it. So it's a high time we have to go for an alternative. So why superbugs? As Lucky already mentioned, what superbug is. So why these are problematic? We human race consider things problematic only when it is problematic to us, right? So if that is the case, then this is very problematic and very concern for a human race in very near future. Because when we dump antibiotics into poultry and livestock, then these antibiotics started creating antibiotic resistance in the pathogens. So when we feed those poultry meat or livestock meat uncooked with the antibiotic residues, those antibiotic resistant pathogens also has a chance of infecting us also. So
So we can find the data here. The smarter bacteria have become highly resistant to most of the antibiotics used already. So here there is a list of gram-negative and gram-positive bacteria, out of which you can find like some species like Enterobacter, Citrobacter, and in a gram-positive bacteria, this Enterococcus species has already developed 100% resistant towards the most common antibiotics used already. And even in other cases, it's like 50%, 20%. And I'm telling you, in very near future, all these pathogens going to develop 100% resistant against any antibiotic we give them. So, you know what is the fate of this? Sorry. This is the cause of it. The deaths attributable to antimicrobial resistance every year by 2050, it is going to exceed the number of deaths caused by cancer itself. So this is very alarming, and this is the high time we have to go, we have to look for an alternative. So what can be used as an alternative? If a problem comes from nature, the solution comes from nature too. We have a medicinal plants for the rescue because they contain amazing bioactives with n number of benefits like antimicrobial activity, antioxidant, anti-allergic, anti-inflammatory, and anti-mutagenic property. Some bioactives also contains antidepressant molecules too, because that is becoming very important, because any infections is always associated with depression also. But still, is there any disadvantage with this bioactive, or only by consuming these bioactives can we combat this antibiotic resistance or any other infections? But no, there are some disadvantages with these bioactives, like poor solubility, poor absorption, and poor stability. Then what can be done to overcome these drawbacks? We have the nano concept for the rescue. Smaller size with bigger impact. How nano can be used? Nano can be used to improve the bioavailability of the bioactives, and it also helps to stabilize the bioactive from a number of physical stress like heat, temperature, light, etc. So one side we have a traditional knowledge, the other side we have a modern concept. And now we are here to combine the goodness of our tradition, wisdom, and the modern concept. So this is the hypothesis we have framed to work on nano antibiotics. Initially, we have identified the phytochemicals of antimicrobial interest based on the literature strength, and also the bioactives which we have extracted using green solvent and physical assisted techniques, physical methods of extraction. And we have also gone for basic plating techniques to confirm its antimicrobial activity. And we have also studied the interaction between these bioactives with the major pathogens like E. coli and Staphylococcus aureus to confirm their antimicrobial activity. And after confirming the right bioactives, we have designed the nano carrier depending on the nature of bioactives, and we have gone to the encapsulation of bioactives into the nano carrier. And we are about to study the release of anti-active ingredients and also its bioavailability. The major mode of action we target is piercing the bacterial cell. As I mentioned previously, the pathogens are very smart to develop antibiotic resistance against any enzymatic actions or protein modifications. So we are just targeting the physical mode of action, that is piercing the cell walls, and to exhibit the antimicrobial action. So the work done so far includes the extraction of active molecules from medicinal plants, from the selected medicinal plants after screening of the bioactives using water as a solvent and physical techniques. And we have encapsulated the active molecules into a designed nano carrier. Again, the nano carrier is natural polymer based to ensure that it is completely organic. And we are into characterizing this nano antibiotics. We have recently confirmed the size and morphology of it, and now we are into quantification of the bioactives that is loaded into the nano carrier. And simultaneously, the molecular docking studies is also under progress to confirm its bioactivity. So the way forward includes the in vitro studies on release kinetics and bioactives. Then when it comes to nano, biosafety always is in line. And we are also into bioefficacy of nano antibiotics on major multi-drug resistant bacteria in vivo, along with ethical committee approvals at varying concentrations, and to assess the humoral immunity and the cell-mediated immunity of the birds fed with antibiotics. So before concluding, I would like to thank our Honorable Vice Chancellor Ma'am and the Dean SPGS for giving this wonderful platform for all our students for sharing our innovations with the industrial people. Thank you.
Thank you. The leftover student from post-harvest processing team, Ms. or Yogeshwari, will be talking on the high vitamin, high nutrient, low saturated fatty acids, low added sugar. Why you are irregular? Why you are irregular? Laws present there, you are not abiding with the time and the rules, Yogeshwari. Be short within 10 minutes.
Yes,
Rules are always for rules. Okay?
Thank you.
You cannot have extra privilege. Start.
Thank you, sir. Very good afternoon to one and all here present here. I am Yogeshwari, Department of Food Science and Nutrition. So I am very honored to be here to present my doctoral research on standardization and evaluation of extruded fortified rice. Why I have chose this topic means in India, most of the people are undernourished, especially children under age between 5 or underweight, and 40% of the women are anemic. So due to this consideration, I have chosen this topic to provide nutritional rich extruded rice to the targeted population. So coming to the topic, we have to discuss micronutrient deficiency at global level. Globally, micronutrient deficiencies spread over 2 billion population globally. Okay, the first public health problem is affecting 1/4 people in the worldwide are affected by micronutrient deficiency. But government of India and government of the world has taken many programs to avoid this problem, but still this malnourishment is still this problem is in our country. So to avoid this problem, food fortification, it is an effective way to eradicate this malnutrition problem. So how the fortification is done means through food fortification. I have selected rice as a staple food for fortification. Because why I selected rice means it is a stable food, it is consumed 60% of the population in India. So without changing people's dietary habit, we can provide nutritional requirement by rice. So what is the technology is available for preparing this extruded rice means extrusion technology. It is an innovative technology in food processing industry. In this slide you can see the single screw extruder. It is an equipment used for manufacturing extruded fortified rice. Actually this extrusion, it is the process of forcing the food raw ingredients into a perforated plate. You can see that perforated plate in the right side. So it is called as die. So through this hole we can force raw material so that rice shaped material will comes out. So once the material will comes out, there is a cutting blade. It will cut into rice shaped grains. So through this extrusion technology we can develop rice shaped kernels loaded with fortificants. So extrusion technology, it is a most effective way for fortification. There are lot of technologies are available for fortification like dusting technology and coating technology. The main drawback of dusting and coating is in that layer the fortificants are dusted. When we cooking the rice, the fortificants are leached out in the water. But in the extrusion technology, the fortificants or nutrients are inside of the grains. So there is no chance for leaving that microorganisms that we are fortified. So these are all the advantages. But you may think of that Is this rice is available in the market? Yes, it is already available in India. There is one global team that is available in India, PATH. PATH means Planning and Appropriate Technology for Health. It is a global team. They are working together with the public institutions to provide ultra rice. Ultra rice means it is fortified rice that is already available in the market. This rice is supplied to children in Karnataka. Here, other than India, some of the countries are manufacturing this fortified rice. China, Philippines, Costa Rica and USA. These are all the countries are already manufacturing fortified rice. Then you can see some of the brands that are available in other countries. So in my research, I have manufactured fortified rice by using broken rice flour. So in this picture you can see ground rice flour plus micronutrient premix. Here I used ground rice flour as broken rice flour, because broken rice, we all know that in rice milling industry byproduct is broken rice. During milling and during polishing, the broken rice wastage, 16% of broken rice are wastage. This broken rice is mainly used for animal feed, and also some goes to the textile industry for preparing starch. But these are nutritionally rich as whole rice. But due to this small size, we are not using this broken rice. So I have used this broken rice as a base material for developing fortified rice. So broken rice flour plus micronutrient premix, that may be vitamin A, iron, folic acid, anything. So in my study, I have used vitamin A, iron and protein. These are all my micronutrient premix. So broken rice flour is mixed with this micronutrient premix. Then it is blended by using blender, then it is mixed, then it is extruded by using that single screw extruder. After extrusion, that rice shaped grains were dried and it is cooled, then it is go for bulk storage and packaging. Finally, you can see the rice shaped grains. So already I said that why I have chosen broken rice as a base material. Broken rice is a cost efficient material, and the rice milling industry can also benefit by using broken rice. Then it is nutritionally good as whole rice. Then there are different treatments were carried out in my study. First one is moringa leaf powder is used for iron fortified rice. Then next one, lentil flour is used as a protein source for producing protein fortified rice. Then next, carrot powder is a base source for producing vitamin A fortified rice. So you can see the picture that iron fortified rice, protein fortified rice and vitamin A fortified rice that have developed in my study. Then you can see a view of packaged extruded fortified rice. Then my research innovation is fortified rice had 100% retention of iron content and 87% retention of protein content and 70% retention of vitamin A content. Okay. So addition of natural form of fortificants did not have changed any sensory acceptability of the fortified rice, and fortificant has higher absorption based on in vivo study I have carried out. Then the other advantage is this fortified rice has very short cooking time, because during extrusion process the rice flour is already cooked. So the final cooking time will be reduced, the main advantage. So this is main advantage for present busy generation. This rice have 7 minutes for only cooking, like noodles we are preparing like that. Noodles are dipped in hot water for cup noodles, I am saying. Noodles are dipped in hot water for 5 to 7 minutes. Like that, this rice also can be cooked. Then it have good storage stability and sensory quality up to 6 months of storage study. Then these micronutrient fortified rice has cost effective and sustainable solution for eradicate micronutrient deficiency in the population. So in my conclusion, I say fortified rice supplementation is the key to solving India's nutritional security problem. So these are all my presentations in international conference by presenting this fortified rice. And I thank Department of Science and Technology for giving funding support to carry my research work. And finally, I thank Vice Chancellor Ma'am and Dean SPGS for selecting this my research to present in front of you all. Thank you so much.
No, stay back, stay back. It's question and session for
Please.
two topics. One is post-harvest technology, and all the four can come, excepting the chief guest. So 5 of you can stand an array of this thing. 4 belong, come, stay there. 4 can be for the theme of nutraceuticals, and she is for a post-harvest processing. Now the question and session is open.
Vareva, give a big round of applause to these lovely ladies. I think fantastic. I'm actually thrilled actually. I'm a new age entrepreneur. I'm entering into agri-tech. I've been an entrepreneur for a very long time. So all of them very, very good. I have one question to the last presenter. You mentioned about fortified rice.
Yes, ma'am.
Can this be done for other dals and all that also? Other broken dals and all that? You get the other pulses, can it be done, or it's only for rice?
Yes, exactly right, ma'am. We can produce protein fortified rice by blending lentil flour into broken rice flour. Yeah.
I see. So that all, so once you develop a fortified rice, you are developing various variants.
Yes, ma'am.
Okay, okay.
I am only presenting 3 variants, but I have developed 10 treatments by comparing natural fortification and chemical fortification. For example, iron fortified rice has been manufactured by natural fortification, chemical fortification. For natural source, I have used moringa leaf powder. And chemical source, I have used micronized ferric pyrophosphate that is used in pharmaceutical industry. So I have compared both.
Okay, very good, very good. And also the chicken person, I think you presented on the, sorry. I just wanted to find out, you know, one question is that, so when you're saying that antibiotics generally is bad for the health, and the I want to understand the core of what you're doing to be able to, so should we eat chicken or not?
Obviously we can eat, ma'am. As I mentioned, it is a very good proteinaceous healthy diet. I'm not here to depromote the chicken. Actually, I eat a lot of chicken.
Oh my God.
But the thing is, antibiotics are really good. But the pathogens are very smart to overcome the antibiotic activity. That is the problem here, because most of the antibiotics given are as a single molecule. It is not a complex. So it's becoming easy for the pathogens or any infective bacteria to overcome or to combat a resistance against that, to create a resistance against that particular antibiotic. So when this, like for example, when there was a resistance for penicillin, the other one came. Yeah. So now there are n number of antibiotics, still the pathogens manage to produce resistance against those. One example we can quote is corona. Initially we were given with 1 dose of different vaccine, and immediately the other variant came up, the other vaccine came up too. So it will continue. There's going to be no end for this. And why we have gone for bioactives, even as Sachin sir mentioned, like when we extract things from a plant based, it is not going to be a single molecule. So it's going to be a complex system. And here I am not using only one bioactive. I have just combined the goodness of 1 or 2 different bioactives of different nature. So that it becomes, I just want to make the system complicate for the pathogens to develop resistance against that particular antibiotic.
Okay, yeah, thank you. That karuppu kavani rice, okay, I just wanted to check with you. White rice and karuppu kavani rice, right? So are you suggesting that we should take more of karuppu kavani and not white rice?
Yes, ma'am.
That's what you're saying. But it's very expensive, no ma? Karuppu kavani is very expensive.
1 kg, I think maybe around 300, ma'am.
Yes, so it's very expensive. So how do normal people take? I mean, the aspiring middle class,
Ma'am,
how do they take?
that's why we have been developing a spoon feed of kavani rice by using nanotechnology, ma'am. We can just take as a pills or as a capsules.
I see, I see. Okay, thank you. The other two presentation, I don't have that subject knowledge, okay? Thank you so much. Thank you.
Any queries from student side? All brilliant students, we hope so. So you'll have questions. Please.
Hi, everyone presented very well, but I have a doubt for rice fortification. Please.
Yes, please.
What about the cooking quality of rice? Because you have fortified with micronutrients. I need to know the cooking quality of your fortified rice. Have you ever tested before?
Yes, cooking quality is an important parameter in extruded fortified rice. Cooking quality is standardized. For cooking quality, water absorption and cooking time is standardized. So cooking time is only 7 minutes for cooking this rice, and water absorption is 30%.
Okay, thank you.
Thank you. So, hi here. Hi. To complement ma'am's question to Mohita, as ma'am asked you, the price of kavani rice is a bit higher. So you said you are going to spoon feed it as a pill.
Yes.
So somehow to prepare that pill, you will be using
kavani as a byproduct.
Yeah. So comparatively, when I am eating 1 cup of rice, when you are preparing one pill,
yes,
you will use the same 1 cup of rice or maybe extra.
No. Why? Because whenever I take that 1 gram of kavani rice, we can get more number of, I can say more amount of phytochemicals from 1 gram. In compared to whenever we can cook this 1 gram of kavani rice to eat. So we can get a more number of phytochemical from that, and we can encapsulate this more number of phytochemical into a nano carrier system. So that compared to cooking, by taking pills, we can achieve more number of phytochemicals, and we can achieve more bioavailability.
Okay, will that be the bioavailability? What I am getting from 1 gram of rice will be higher than 1 cup of rice. You're meaning like that? Okay. Then to yes, please. You said the broken rice are already cooked. Yes. So already the rice is cooked, and then we are extruding, and the product is prepared. So once again, when I'm I will be steaming the product, as you said, for me to consume. So there will be no degradation of any carbs and nutrients while I'm going for like 2 cooking, I think. Yeah, exactly, you are right. I have formulated this micronutrient fortification level based on extrusion laws, based on cooking laws, based on storage laws. So these losses are compensated in fortification level. So this is formulated. Okay, but somehow there will be a loss of nutrients. Yeah, exactly. Okay.
I have a question for you. The extruded rice. Okay. Have you tasted your own product?
Sorry, sir?
So you have
Yes, sir.
come up with some products.
Yes.
Have you tasted it?
Yes, sir.
Will
Sensory
you accept
evaluation.
it? Will you accept?
I'll tell you, sir. Sensory evaluation was done for this extruded rice. Variety rice preparations and biryani rice was prepared, and sensory evaluation was done by 10 panel judges. And the score value of 8 was 8 out of 10.
Okay.
That is not mentioned in the slide, but it is done.
No. If you had presented that also,
okay,
it would have been advantageous, right?
Okay, sir. Thank you, sir.
Hello, question from here. I have a question to Ms. Yoheshwari.
Yes.
Regarding fortification.
Okay.
Have you used that moringa powder and carrot powder and all used for fortification? You
Okay.
use the powder acid. Yes. You mix with the rice powder.
Yes.
Instead, can we extract that if you go for iron fortified rice, can we extract that iron nutrient from moringa powder and then mix it, because it will reduce the bulk, it will be more nutritionally acceptable by.
You are right. We can extract that particular compound in that, that is a good work, but it will take a lot of time to complete my study. So I have powdered and added. Because standardizing extrusion conditions take long time. I have done whole 1 year for standardizing extrusion conditions. So if we go for extraction means it will take some more time. So that I didn't done.
Thank you.
Thank you.
Hello. Yeah, it was a very good presentation by all the 5 innovators. My question is for Sir, Sachin sir.
Yes.
Sir, it sounds like a strange question though, but you are from the same IT field, and you know it is a race against time working in IT field. So is it possible to replace food by the developing nutraceutical chemical like tablets? Like we can have 1 tablet for carb, 1 for protein and all for minerals, and have a glass of water and go for work. Is it possible, and is it possible in the upcoming generations?
Might be possible, but I wouldn't enjoy it. I will still enjoy my food. I love my food. So technically, maybe at one day you can replace it. But again, the satisfaction you get out of eating good food, which is a sensory part, is a lot more important for us as a culture. So yes, but getting everything what you need for your body, you might be eating a lot of capsules. Yes. So might not be practical, but with nanotechnology, you never know.
Okay. Thank you, sir.
Thank you.
And thank you, Sachin, and you can take your seat. And first of all, I think a big applause to Mr. Sachin, because he flew down from Hyderabad in the morning, and now he is going in the evening. He is such a busy person, but I think he just, when I requested him and he asked who are all the audience, then I told students, then he said, okay, right, because an IT professional turning into a top agricultural entrepreneur, an IT guy turning into agricultural entrepreneur means it's a success for agriculture. That's what I can say. Many people from agriculture is going to other sectors, and he is coming from IT to agriculture. So I think feel proud that agriculture has actually attracted Sachin, and now he is one among the leading entrepreneur on agri food space. So thank you very much, Sachin, for your time and as well as for the interaction you had with the students. So now I think almost we have done this today morning's program. And thank you very much for the wonderful presentation by these 5 PhD students. And what I did is, now I have with me one Mr. Rajan Kanagasabai, and he is again from IT sector, and he has a huge experience on dealing with students or technology development, and as well as what he does is he can evaluate people and also he can groom people to the next level. He doesn't know anything about this particular conference. Today morning I called him, students are going to present, just come and see and give your feedback. If at all anything can be improved from their side, because always improvement is a continuous process, and you are all students. Even today also I am learning. Learning is a never-ending process. So I think I have invited him. Just I'll give 5 minutes to Rajan Kanagasabai, and probably he'll tell exactly what about this presentation, and any improvement opportunities are there, probably he will highlight. Mr. Rajan Kanagasabai, please, for your comments.
Good afternoon, everybody. And I know I'm standing between all of you and lunch, so I'll try and keep this very short. Thanks, Dr. Lakshman. I think it's amazing honor to be in front of all of you all, especially the students. As Dr. Lakshman introduced, I am from the IT industry. I've been the global vice president for Hewlett-Packard, companies like Mahindra, Satyam, so on and so forth. And of course, now I'm more into my own entrepreneurship. And I deal a lot with agriculture, not from the agro sciences front, but more on the IoT, the artificial intelligence, the drones, those kind of stuff. And that actually forces me to be actually seeking a collaboration with Dr. Lakshman and people like that. To keep this, I mean, I was really blown by the kind of depth and the kind of details that have been presented here. So again, a huge round of applause for all the 5 participants. What impressed me really most here was the flow and the thought process and the research. Obviously, there are, it's very, very obvious that the guides here are extremely strong behind these people. And the innovation and I mean, it's one thing to innovate, but it's another thing to present it in a sequential flow, which kind of, impresses everybody. So I think the thought process, and given the time, I think each one had a very little time. And to convey an important message, like for example, the one which spoke about eradicating the nutrition deficiency in the country, to talk about such a huge subject in few minutes takes a lot of research, and hats off to you for that. One small advice, I mean, I wouldn't even call it an advice, it's kind of a suggestion if you may. I think all of you are excellent, especially in your subjects, agro sciences, but when it comes to the next stage of your life, that is presenting it to the outside world, the presentation becomes extremely important. Yes, the quality of your presentation, especially the student who spoke about the chicken, was very, very innovative. I mean, the conversation between the country chicken and the other one, it was very thought-provoking, because if you recall, when Apple iPhone 4 was launched by, I think it was by Tim Cook, it was exactly on a conversation between Android and iOS. So I think it's very nice that you could pull that off. Coming back to that, I think, as I said, your presentation on the marketability of your products at the end of the day is very important. Like you have very good data on your technology sides, as I said, but it has to be, I mean, for a commoner like me, who's not an agro sciences person, for me to understand the marketability or the benefits of this to the various domains, if that is one of your key areas which you present when you go out to the outer world, that becomes a very, very key piece of information. We call it metrics in the IT world. That becomes a very key information for companies to actually understand what this innovation of yours could do and what could be their return of investments in a period of X number of years once they, adopt you or your technology or sponsor your technology. So that is the only possibly single piece of suggestion, if you may, that I would possibly have. But otherwise, again, thanks all for your time, and thanks again for the lovely opportunity.
So thanks, Mr. Rajan. Now, my heartfelt thanks to all the students who have attended this particular program, and in a way that I think that, and very good questions, and thank you very much. And everything what we do here is only to take you to the next level. That's the whole idea, build confidence. And create a good career to you all. That's the whole idea. Whatever we do in this university under the guidance of our Honorable Vice Chancellor, it's all about taking you to the next level. And thank you very much for your presence. Now that morning session, actually we are closing the morning session, and with your request to all the students, please reassemble by around 3:30 p.m. Today. Because so the simple reason is Honorable Vice Chancellor want to address you, and the 3:30 to 4:30, just 1 hour, a final session. This is exclusively on disruptive technologies or emerging technologies. And there will be a one lead presenter, and followed by 3 or 4 students will be presenting on that final session, followed by the valedictory session, where I think the presenters will be awarded for their good contribution in the couple of days. So I think I request all the students, and thank you very much for your support. And one last request is, please reassemble by 3:30 p.m. in the same hall. Thank you very much.
of students, their innovation. And after that, the valedictory session will start, in which our Vice Chancellor will be addressing you all. So this is a program, and we will try to finish maximum by another, by around 5:30 we are trying to finish. That's the whole program for PhD. Right, so now I have the pleasure of calling Mr. J. Krishnan, COO, Trinity Wireless, and who will be delivering the address. I request him to the stage. And also I request the student innovators to also occupy the stage. And now I request Dr. Agro to honor the guests with bouquet and as well as the memento. And probably you can introduce the guests to the audience, Dr. Monica, please.
Mr. J. Krishnan is the COO and co-founder at Trinity Wireless, an award-winning product-centric company focusing on fleet management, mobility, and IoT solutions. Trinity has clients in 15 plus countries with clients in both private and government sector. Trinity has recently won the prestigious award for excellence in location-based services in Australia. He is also the COO and director at Agro Technologies India Private Limited, a pioneering India-based full-service provider for outsourced product development, mobile applications, web applications, and design solutions. The technologies has fully owned subsidiaries in US, Canada, UK, Singapore, and Hong Kong, providing cutting-edge solutions to both corporate clients and innovative technology companies in India and abroad. He graduated his BE in Electronics and Communication Engineering from CIT and MS in Electronics from Dalhousie University, Halifax, Canada. He has published various IEEE papers and journals in the field of robotics and embedded systems with close to 20 plus years of international experience in embedded systems, wireless networks, agile approach, and software development. He is also primarily focused on product development, R&D, architect solutions to overcome technical and real-time business challenges. His interests are towards inculcating creativity, problem solving.
For the introduction. And good afternoon, everyone. Good afternoon, everyone. I know this is a death hour, especially after lunch and after a wonderful conference and expo headed by the Vice Chancellor and the Dean, Dr. Lakshmanan. I think that really deserves a round of applause. Can we start with claps for them? Yeah. So I think that hopefully wakes everyone up now. So before I start into the topic, when I was meeting with Dr. Lakshmanan, and he said, please take a tour around the expo. And myself and my colleague Mr. Rajan, we went around and took a look at what was being displayed. And it was very interesting for me because I'm not coming from this specific background. But at the same time, it was very intriguing also on the possibilities that lay ahead of us. And 6 sessions, and possibly in the future conferences, we will see more on the sessions related to IoT, drones and things like that, equal to crop management and other things. And that's the whole idea as we move forward. So quickly moving on to the session. So today I'm going to be talking about enabling smart agriculture using IoT, AI and drones. The whole idea is to give you an overview, give you an understanding of what are the potentials, opportunities that lie ahead, and how as students you can benefit by leveraging technology into your specialty, which is in the space of agri-science. I'll try to map both of it and give you a flavor of what's ahead for us. So a very short 4-point agenda. Just quickly speak about what we do as an organization, just for the context of when I'm giving certain examples and case studies. So Trinetra is around 10 years in existence. We are part of a group of companies which is 30 years in business, and we are located in Coimbatore, our own Coimbatore. We are headquartered, and we have offices in Chennai and few other locations. The whole idea and concept is that we are wanting to be a very dependable technology and product solutions provider, harnessing technology, digital innovation, and taking it to the stakeholders through remote sensing, IoT, drones, and things like that. So we have a lot of assets that's deployed, assets in the space of agriculture also, and non-agriculture as well. So quickly moving on to Agriculture 4.0. I'll not go too much into this because I think from yesterday to today, I think a lot of them would have spoken about it, but in a nutshell, I'll try to put across what really Agriculture 4.0 means or smart agriculture is. So before we move into that, I think one needs to understand that there is one is the expertise, which is in the agriculture space, and then there is the opportunity, and then there is the technology. The technology is largely facilitators that is helping you to take your products or services to the market. Very simple example before I go into this is how, let's say, an Uber revolutionized how we started using taxi services. It changed the whole way we use taxis, right? No longer negotiating with taxi drivers. Taxi comes to your doorstep. And that's the power of technology. Helps you to facilitate things in a much more faster way. There the advantage was the consumer also knew the technology very well. The consumers are people like us, knew the technology very well. Let's take Amazon for example. It's revolutionizing the way we do online shopping. Now, I don't know when I last went to a mall or when I last went to a garment shop. Now, everything is on a click of a button, and to make it even better, if you don't like it, you can return it also. Again, here I think the technology was easily adaptable. But the gap that we kind of foresee when we do market research is that in the agri space, while there are a lot of expertise in terms of students, in terms of scholars that is using in the agri space, but the challenge lies in understanding the technologies, the awareness of the latest technology or the deep learning technologies that is available, and how to actually inculcate it into your area of space. So the main thing, if you can see, I think it focuses on lack of access, availability, understanding this technology. Now this gets even more challenging when you go into cutting edge technologies and the emerging technologies like IoT, Industry 4.0, Agriculture 4.0 and things like that. So the whole idea is how do we marry your domain expertise in the space of agriculture, using technology as a facilitator and then taking to the market, helping you to scale up. That's the whole idea. And this is where I feel there is a little bit of a gap, and a gap that can be easily overcome by just understanding what is available for you. And the whole focus of this presentation is to give you a flavor and an overview at a high level on how such technology is used in the agricultural space. So smart agriculture. So I think this it's a very crisp slide, and here the focus is going to be largely on Internet of Things, which I'll be commonly referring to as IoT. Right? So how do you make agriculture, which is a very, very important cog in our GDP, in our industry, in our business ecosystem, in our food chain, how do we make it even more scalable, even more sustainable? How do we take it to each and every corner of India? Right? So one way of doing that is to smartly adapt to the various sensors that is available through IoT, and then taking a lot of data, mission information, agricultural information, weather information for decision making. And what's interesting is that the global analysts and the experts are saying that this is really going to be booming. It's already booming, and you can see most of the governments, especially the governments of India, also spending a lot of effort and thrust towards agriculture. So it's expected to hit a whopping $6.2 billion in the last year itself. So that's the amount of scope that is available and the opportunities for you as students in front of you. So in a nutshell, there's basically 5 components in Agriculture 4.0. You have all these sensors like the weather sensor, the crop monitoring sensor, the health sensors and things like that. And then you also have opportunity to remove a lot of hard manual labor through using robotics, which helps you to automate tasks, right? Then of course you have assets or devices or trackers that is going to help you to get data, track the information, track maybe livestock, and then understand the whereabouts, the positioning and things like that. But all this is possible only if you have better connectivity, and that's where I think the telecom revolution has also helped us a lot in terms of availability of 4G networks, 3G, 2G, SIM cards. So many other kinds of communication networks are also coming. You have things like Sigfox is a new technology, LoRaWAN is a new technology, so many things that are coming up. So the advantage of this technology that we can leverage is sitting in your home, sitting in your college, you can actually start monitoring farms and fields very far away in real time. And of course, why do we do all this? We want to take informed decisions, for which we need to do a lot of analytics on the data that we have. And how do you differentiate between data that is informative versus data that is just data and not really too much of informative, right? So eventually leading to better decision making. That's the whole idea of all this data collation and analytics. So we also want to touch upon a little bit of artificial intelligence as well. Because once you have all this information and once you have all this data available at your application or doorstep and things like that, one is looking at the data, taking information. The other is how do we also start forecasting? How do we also start predicting certain things? And this is from one of the big 5 consulting firms. And these are some of the use cases around the agricultural space alone, in terms of how AI can be implemented, in terms of predictive, in terms of how your crops can grow, what is the intelligent spraying methodologies, where you have good plants versus weeds and things like that. Crop and soil monitoring is another important thing as well, because that's what determines the quality of your produce as well. There also we can do a lot of machine learning, artificial intelligence and diagnostics as well. So this is just a nutshell in terms of what is the potential capabilities that you have. And any space that you take, I think IoT, AI, there's a lot of scope and potential. So I'll quickly move on to actual case studies. Some of these case studies you might have seen it in your previous presentations also. And some of it are actual case studies that we have done at our organization, which we wanted to also share with you. Let's start with the most simplest use case that is available, but possibly very, very important as well. Monitoring climatic conditions, right? So weather stations, what is the sensors that picks up weather information, weather data. This also has a very important say in terms of, when do you want to water your crops? If rain is around the forecast, do you really want to water it and things like that. So this starts with the most fundamental aspect of monitoring the climatic conditions, and then eventually leads to how well you can utilize your most important resources like water. So that's a very simple use case. This is also fairly mature in terms of greenhouse automation. This is very similar to like your smart home automation, right? You click of a button, you turn on certain things, you switch off certain things. So here also you can get atmospheric data, lighting information, temperature, soil information. And based on that, you will be able to take decisions on what should be the next step for your greenhouse. So again, simple use case, but I think these are the basics that we need to go through. So this is very interesting, crop management. And in fact, there are a lot of sensors that is available now on the market where you can actually have sensors implemented at the soil level, which measures a lot of things, like right from moisture content, soil. You can even go to more complex aspects like some of the nutrients and things like that as well. And then all this even relating to overall health of the crop. So essentially the fundamental layer, if you see, a lot of data you are collecting, accumulating all this data, sending it in real time, doing the analysis, then taking the decisions. So that's the overall process so far that we have seen. We'll start going to slightly more complex use cases also. So far we saw about crops which are all largely stationary in nature. Now we can also extend this into little bit of mobile, animals, assets and things like that. Here I think the whole gamut changes because it is no longer confined to a single space. And this is where technologies like drones also comes into existence. Let's say you have a big field and then cattle is grazing. You also need to mark areas where they're allowed to go and not allowed to go. And the moment there are some cattle that goes beyond a particular segment like a geofence, then alerts can be triggered in real time. alerting the people to come and take some actions on it. Other examples is depending on the temperature, you can also do tagging of animals. Depending on the temperature, you can also look at what is, if any animal is infected, so maybe you need to segregate that animal from the herd and things like that, because we have heard a lot of cases where you'll have to unfortunately cull animals, birds and things like that. So those kind of livestock handling situations can also be done. So drones, I was speaking to a gentleman and he comes from the drone industry, and he was saying how so many agricultural drones is taking traction in and around Coimbatore, and largely used for surveying purposes, also used for watering purposes. Imagine the amount of work that is going to save and time it's going to save for humans to do this. You're talking about acres of land here. How do you even cover it? How do you even survey it? So drones actually can really help in such situations, and it's said in studies that it's 6 times much more effective, time saving, cost efficient than actual humans doing it. Now moving slightly away, we spoke about stationary vegetation, then we went into animals. Now let's take an example of a sugarcane industry where they have something called cut to crush. And these are actual case studies that we work with industry leaders in this space. And here the use cases, the moment a sugarcane is cut, they have a KPI defined, and it has to reach the factory for the crushing process to start, so that the produce of the sugar is much more better. And again, it's very, very vast fields. It's in really remote locations. The sugarcane gets cut, loaded into a tractor, then it gets loaded into another lorry, then it comes to the factory location where the crushing process starts. So you could see the amount of changes that happens. And how do we measure and monitor all this? Again, through IoT devices, where the tractors can have IoT devices, the vehicles can have IoT devices. So on the moment we are able to cut, load it into the tractor, your clock starts ticking. You start looking at aspects of when it's starting, and then you can again mark zones, fences, and it enters the factory location, you get alerts. So you can actually prepare for your production much before the sugarcane actually arrives at your factory gate. So that is the amount of insights and forecasting that you can do much before it actually reaches your factory location. So here are some of the use cases, and what's interesting is the saving. If you look at the cost saving, it's around 30 to 45% of cost saving. On-time delivery is also increasing quite drastically by 25 to 30%. And imagine acres of sugarcane, and imagine the amount of crushing process that has to happen. So it really runs into crores of rupees in terms of profitability to the organization. Another similar example is, let's say you have your perishable products. It can be your fruits like apples, strawberries, other kind of fruits that requires little bit of controlled temperature, or it could be frozen stuff, or it could be dairy as well. Dairy is your milk, cheese, those kind of produces as well. And here also the role of IoT is that not just location tracking, but also in terms of monitoring the temperature within the compartments. So if the milk has to be at a certain temperature, then it can be set, and the moment it crosses beyond the temperature, then alert starts to happen. So again, it is very, very proactive. With the alerts, a lot of business rules can be set in place as well. So again, in terms of savings, you can see in the right hand corner, quite a number of savings over there. Customer satisfaction eventually is very high when we do this. Again, it's very simple technology, data acquisition, set business rules, thresholds, you're able to take decisions out of it. So this is also very similar to what we discussed, so maybe I'll not spend too much time on it in the interest of time, but these are like cold room monitorings, where you have the applications can be again your ice creams, your frozen food, or maybe you can even extend it slightly away from agriculture. It could be like pharma kind of things as well, where it has to be really controlled. Again, here this is a stationary environment, multiple compartments, you can look at temperatures, take decisions out of it. So these are some of the use cases and the potentials, just for all of you to understand that it's a very, very vast scope, and when you marry this technology with your agricultural agro sciences expertise, then I think the amount of time saving, cost saving that you can bring to the table is tremendous without putting in too much of effort. The key is without putting in a lot of effort. This is just a pictorial representation of how you can actually do a farm-based monitoring. Multiple sensors goes in, then you have a device that you can control and transmit the data. It comes to the servers, and you can actually see it on a mobile app, and you can actually trigger when your motor should start watering the field as well. So all that can even happen just sitting remotely. Another thing in the poultry segment that is really catching up and has a potential is looking at livestock and looking at all the critical parameters like temperature of the bird, size of the birds. You can use cameras to determine the size of the birds, temperatures to look at the what is the actual temperature of the bird and things like that. You take a lot of collation of this data, and then you'll actually see where the exceptions are, and take decisions in terms of segregating the infected ones from the live ones as well. So again, imaging and things like that. So very interesting use case that we had yesterday when we were speaking to one of the poultry transportation companies is that they wanted to know what speed their vehicle is going. Each time a harsh braking happens, there is damage in the container. So small things like that has a major impact. So it can go to the granular level of getting enough information and data, which has actually got a massive impact in terms of how your produce comes out. This is just a pictorial representation of how things work. I think most of you would know this by the end of the second day of the conference. But yeah, data collation comes to our device, sends it through satellite, 3G, and then you can get it in the application on your mobile phone with an integrated dashboards. So I think why do we do all this and what is the benefit for the students? I think that's extremely important, and I think I understand that here there's a mix of UG and PG students as well. So the benefits are multifold as far as students are concerned, and agriculture I think is more and more becoming a hotbed in terms of focus for many large conglomerates and also for the governments as well. So if you look at where it starts is to ensure that how do you again combine theoretical know-how, quality research into proper field work so that the exposure level are really very high. So how do you take it to the market? I think that's the whole question here. So exposure is the first thing, and I think now with TNAU taking such initiatives, it's a great opportunity for all of you to explore on those things. It also helps you to go into the sphere of entrepreneurship and innovation. Start something on your own, right? I'm sure that's a dream for many of you, and it's not very difficult, especially in this current age of funds available, support from the institution, support from the university. It's definitely possible, and a lot of large companies are really looking forward to working with agile startups, working with companies from incubators and things like that. So it really opens up a plethora of opportunities in the space of innovation and entrepreneurship as well. And of course, I think if you have a quality research and quality amount of work done during your theses or during your UG and PGs, I think if you again understand the technology capability, that's what industries are looking for. And there's a lot of opportunity not just within India but outside India as well, with very good grade structures and things like that. There is a clear need and demand for agri scientists, agri students, agri sciences, but with little bit of technology know-how, then you become a really powerful quantum altogether. So these are some of the benefits for the students that you can gain out of it. And how do you also actually make best use of it is organize and take part in conferences such as these, where you display your work. A lot of hackathons happen in our city as well. I think taking part on that itself will give you a lot of exposure. A lot of companies are offering internships as well, so you should look out for such things as well and see how you can get real world industry exposure as well. Look for identifying constraints in your segment. It could be very simple, small constraint also, but I think it's very important to look at the constraints and see how you can remove the constraints. I think all the startups that's raising funding is actually just trying to spot a problem, fix the problem, make it easier, faster, cheaper to ensure that the solution is found. So I think the third point is quite important. I think the other things I think we spoke during the course of the discussion as well. adapting and aligning to emerging technologies in the space of agri-sciences. I think again agri-science with the emerging technologies, I think that's a space where we can really do well in terms of taking it to the market. And of course you'll have to be very, very focused on what is the demand at the moment that we need to focus on. Timing is about everything, right? So I think we should look at what is the market requiring now. So I think that's very important as well. So these are some of the focus areas for students to look at. The previous slide of course talks about the 3 things, but how do you do it, how do you get the exposure part is in the next slide. So I think the conclusion is that I think there's lots of data, lots of things like that you can do, but I would want you to just focus on the first 2 lines of the slide. The potential that the technology in IoT space, the AI space has is tremendous. I think changes are going to be permanent, it's inevitable. How fast we are able to embrace the change, adapt to it, learn from it is where I think the success lies, and how consistent we are in terms of being a better version of ourselves compared to yesterday. I think that's where I think the benchmark should be, not comparison with others, but comparison with yourselves in being a better person compared to yesterday, day before, and things like that. So with this, I thank you for the opportunity to TNAU, and if there's any questions, I'll be happy to take.
Yes, students, a lot of engineering students are here, so I think probably you can just clarify your doubt. So because this is again, whatever the subject he has touched, so all are very much important. Probably you may not find directly in your curriculum, but these are the technologies, and these are the things normally, this is called as School of Finishing and Grooming. Whenever you talk so much of your theoretical knowledge, these are the technologies will improve your soft skills and give you more confidence. And here I think whatever he touches about is all about sensors and other things, and how it can be interlinked with our own agriculture. And now I think one thing is very clear. There is no separate domain like agriculture, engineering, sensor. So everything, how you are going to create a link between all these things with the whole idea of doing the agriculture in a better way. So that is all about. So I think probably if you throw some more questions to the speaker, he'll answer. Thank you.
Sir, I have a question. Actually, you have IoT and IIoT, right, sir? Industrial IoT. So, you think in which the criticality of the data is high in IIoT or IoT?
A very interesting question. Thank you for asking, madam. I think the criticality of the data is, in my opinion, is almost the same in both. The yardsticks are different in terms of what you do in IIoT and what you do in agri-sciences or agri-field. The yardsticks are different because in agri, I think you have a lot of external factors, external parameters, temperature, you have things like weather and things like that, which is quite volatile. It changes quite a bit. Whereas in IIoT, it is largely a controlled space, a controlled environment, where the external factors are quite low and the mission parameters is what that keeps varying. Yes. So that's the main difference, but the criticality is very, very important for both.
Okay.
Yeah. I hope I answered. Yeah, thanks.
Again, by considering the importance of this IoT, do you think that this is a time that you can just at least introduce a small course or a team teaching in IoT so that the students' marketability or their knowledge, because now you say that business houses or companies, they look for such skill from the students. I think, do you think that this is a time that students, at least in a short course or something, some diploma or something, it's a time that, because we are as a postgraduate dean, I'm seriously thinking, why don't we introduce postgraduate diploma in 3 months or program for them to understand this IoT. Do you think that it's a need of the day?
Thank you, Dr. Lakshmanan. I think it's a very pertinent question because I think when we hire team members for our office, one thing that we constantly have a challenge is the pace at which the technology is moving versus the amount of students coming out of college. It's very hard for the students to catch up. It is not their fault. It is how the technology world is moving so fast. So definitely I think, and we have tried this in few other institutions as well, and it's worked out well for us. We also, like I said, we also offer internships as well. So this kind of a 3-month course where students get exposed on the architecture, the basics of how IoT works with specific to agriculture is definitely something that we should consider because I think it will help the students also to be better positioned when they are about to go for their next jump in their career. It's very, very important aspect and something that can be easily worked out, Dr. Lakshmanan. Yeah.
Thank you. Because I think IoT is a relative term. IoT means sounds differently for somebody. For an engineering student, it sounds differently. A student who is doing in textile technology sounds differently. A student who is doing agriculture sounds differently, but agriculture is the most tough topic. Correct. Because we are working with an environment which cannot be controlled. It's not like working in an industry. So I personally feel that we have to just fine-tune this IoT that suits to our own student, which will at the end of the day, I consider as a soft skill, that will make the student to face that reality in a better way. And when they go for in a corporate sector, when they are positioned, because everyone wants to check their knowledge on this interneting of things. It's nothing but how to create a coordination. And so I always used to remember one Tirukural from, that is, Yerinum Nandram Erividudal Kattabin Neerinum Nandradan Kapu. So those days, Erini means ploughing, Nandram Erividudal means fertilizer, Erinum Nandram Erividudal Kattabin, that is weed management, Kattabin Neerinum, Neer means water management, Nandradan Kapu means pest and disease management. And Thiruvalluvar, those days, he clearly explained the importance of all these things. Already he has done that IoT, Internet of Things, but now we want to do that IoT through sensors and other things. So probably I think it's good time that we have to think about sensitizing the students on this IoT platform. And thank you very much for your time taken in just enlightening our students on this particular topic. At some point of time, certainly we'll have a discussion to take it forward. Thank you.
Thank you. Looking forward. Thank you so much. Yeah.
Machine will rule in 2025. Machine will rule in 2025. Half of workforce will be through automation. Labor crisis is an alarming experience to the farmers. Here, the weed also causes yield loss. Unmanned aerial vehicle allow the site-specific efficient methodology, image acquisition. In further, works in specific on algorithm. With these complicated setup, machine learning will be ruling the roost and double the workforce from 72,000 from 4,000 in 2019 to 2018. With this introduction, we are pleased to invite Mr. R. Arokya, Infant Paul, to talk on drone, an advanced tool for herbicidal weed management in direct seeded rice. Now, Arokya, Infant Paul, will take only 10 minutes to complete.
A warm good evening to everyone. Myself, Arokya Infant Paul, second PhD scholar, Department of Agronomy, and my chairman is Dr. P. Murali Arthanari, Associate Professor, Department of Agronomy. First, a drone is a noble employment to the man. Based on this concept, we have one idea is drones, an advanced tool for herbicidal weed management in direct seeded rice. It's a technology readiness level 1. We have only idea. Why in rice? Because rice is a major important staple food crop in worldwide, and in more than 50% of the human people consuming rice in their food diets. And in India, rice is cultivated in an area of 45.1 million hectares with a production of 124.3 million tons. In India, rice is cultivated in Uttar Pradesh, West Bengal, Odisha, and Tamil Nadu. In India, the rice export is now increasing. See the statistics. In that, last year the rice export is major. It around 17.72 million tons, and it consumes 36% of the total export share. But in the conventional method of rice transplanting is more laborious methods. On the other sides, the labor availability is very scarcity. According to the Food and Agricultural Organization 2020 report says the agricultural labor force declining from 800 million people to 600 million people, and the labor availability is declined to 25%. So we have to address these issues. And on the other sides, the Ministry of Labor and Employment is reported that the labor wages was increasing. So during last 7-8 years, the labor wages was increased to 213 rupees to 338 rupees. It's recorded 37% of the labor wages hike. And for women laborers, the labor wages increased to 177 to 275 rupees. It recorded 36% of the women labor wages. But in the conventional method of transplanting is consuming both labor and it's increasing the cost of cultivations. So nowadays the area under conventional transplanting is converting into direct seed rice. So we can save nearly 88% of the labor and 15% of the cost for sowing alone. But in direct seed rice, what is the major problem is weeds are the major problem, because weeds and crops emerge simultaneously from the germinations, and the weeds compete with the crops for all the growth limiting factors. So we have to address this issue. Generally, the weeds create 40 to 50% of the yield losses compared to the remaining pest, and in direct seed rice, up to 90% of the yield losses was recorded. So we have to focus and control the weeds during the critical period for getting the higher yield. For this, there many conventional practices are followed. One is hand weeding and mechanical weeding also is there, but we have to focus, because it is a labor scarcity issues. We have to focus on labor problems, and we have to develop some alternative solutions, that is herbicides. Herbicide is one of the alternative methods. It can be applied by manual knapsack sprayers, but again it is a problem because of the unavailability of labor. And the application of herbicides in the puddled field using the manual labors, it is a more labor tragedy process. So we have to focus the alternative methods. To considering these issues, we have some solution, that is drones. This innovation is a disruptive type of innovations. So we have to provide and give the solution to the existing problems. The drone is a flying device which can fly with the help of GPS and automated coordinates. Application of herbicide using drones, we can save the time, and timely operation is possible using drones. We can save time nearly 20 to 30 minutes. It's enough to spray 1 hectare of land. So we can save the time. And recorded the uniformity in spraying compared to the manual application of herbicides. The application of herbicides using drones, it's reduced the labor tragedy. So no need to enter the laborers into the field for application of herbicides. And it recorded nearly 80 to 90% of the weed control efficiency compared to the manual application of herbicides. Application of herbicide using drones, we can save nearly 165 megajoule per hectares of energy. We can save it, and we can save the water nearly 440 liters per hectares. It's around 88%. And compared to the manual application of herbicide, we can save the cost nearly 27%. It's around 550 rupees. But nowadays, everyone focusing on the precision farming. So applying the herbicides where the weeds are located. That's a new concept. First, we have to take the images of the fields by using the sensor-mounted drones. It will capture the field images, and after that, using some softwares and sensors, we have to develop the weed mapping. It will give the distinction between the crop and weeds. Based on the data, and we can identify where the weeds are located. Based on this, we have to provide the input file to the drones, and it can apply the herbicide on the particular location in the site-specific manner. And the impact of innovations, quantitatively, this innovation helps to reduce the water, time, and energy, so we can save all those things, and it can save the tragedy of the laborers. It reduces the tragedy of the laborers, and qualitatively, the application of herbicides in the precise manner is possible, and it is a potential tool for the future in the precision farming, so we can save the soil health and wealth. And scope and utility of the innovations, the union government is now focusing on the modernization in agriculture, so the industries can take this kind of idea, can utilize this information's technology, so they can transfer the technology to the farmers by collaborating with the farmers organizations and hiring centers, and it provides a lot of opportunity to the younger generations for the developing the new startups. And future line of works, developing some herbicide formulations especially suitable for the drones. It can be a new scope for the development of herbicide formulations. From this, I conclude that the future agriculture is fully depends on modernization and mechanizations, and I hope a drone will fulfill both things. Thank you. Thank you everyone for the wonderful opportunity.
Thank you, Aurokya Infant Paul. And the next on the B line is Ravori Sai Prasad. He had made a big leaps to detect, determine, and to have a scope for a wide range of operations in his design and development of robotic harvester for capsicum annum under protected cultivation. Now, Ravori Sai Prasad will be presenting on its robust mechanism.
A warm evening to one and all who all are present here. Myself, Ravuri Sai Prashant, pursuing PhD second year in the Department of Formation and Power Engineering. I am doing my research work under able chairmanship of Dr. A. Surendra Kumar, Professor and Head, Department of Formation and Power Engineering. Coming to my innovation, I am going to do research on design and development of a robotic harvester for capsicum under protected cultivation. In today's world, robotics is an emerging and upcoming technology which occupy more space in technology development. And it will reduce human burden in all the sphere of activities like medicine, agriculture, and allied sectors. It is a need of our and order of the day. Coming to my sort of innovation it is disruptive business model. In my innovation, it will leverage more vistas for harvesting easily and free burdenly. Drudgery reduction is the key point in my innovation. Coming to capsicum, how many of you tasted capsicum curry here? How many of you like capsicum? Nowadays, if you go to any restaurants or hotels, in all the food cuisines we can see capsicum. Coming to pulao, fried rice or fast foods, everywhere we are seeing capsicum. And why it is having high demand nowadays? It is having high nutrition values like it is rich in vitamin A and C. It is also having high rich minerals like calcium, magnesium and potassium. Not only nutritional and mineral values, it is having high health benefits also. Like it is good for health and it lowers the cancer risk also. Coming to capsicum data point. In India, India holds 13th rank in export prospects. The major countries which are getting capsicum export from India are UAE, Bangladesh, Qatar, Oman, etc. And if you see the statistics on the left side, here I have presented the past 6 years data of area of production and also of capsicum, similarly total production and area. If we see the graph both, there is a significant increase from past 5 years. And now, according to 2021 and 2022 estimates, the total area of capsicum cultivation is 38,000 hectares. And the total production is 5,63,000 metric tons. And these are the major states of capsicum cultivation based on the productivity. Like capsicum can be grown all over the world. We can see from Tamil Nadu to Jammu and Kashmir and Madhya Pradesh to Meghalaya. These are the top 10 states in the productivity order. And in Tamil Nadu, major district is Krishnagiri, where we capsicum cultivation is grown in protected cultivation. Let us see what are all the capsicum physical properties. Like capsicum is basically a perennial crop and its duration is around 8 to 10 months. in protected cultivation, the capsicum is grown in indeterminate forms and its height varies from 1.8 to 2.5 meters. And the type of sowing. In protected cultivation, capsicum is sown like after the 45 days of seedlings, those are transplanted on the raised beds. And then sowing pattern. It can be sown in single row or paired row methods. Coming to harvesting stage, we can see like from the 80th day of transplanting, there will be continuous harvesting till the end of the crop, like until 10th month. In normal forms, we can say like at the end of 3rd month till the 10th month, around 7 months, we will be getting continuous harvesting and continuous yield also. And if we see in protected cultivation, the yield is 60 tons per acre. And the average number of capsicum per plant will be 25 to 30 per whole season. And the major colors of capsicum which are grown in India are green, red, and yellow. And still, the conventional practice of harvesting is manual harvesting only. Let us see what are all the challenges faced in manual harvesting. The first thing is repetitive harvestings. As I said earlier, from the 3rd month, we will be getting harvesting continuously till the 10th month. For every 4 days, we will be getting harvesting. And also, for every 4 days means, for 1 day, we will be getting around 1 ton per acre. And for every, in a whole season, we will be getting around 50 to 60 harvestings. And it is more tedious work. And then excessive labor. For harvesting 1 ton of capsicum, nearly we require minimum 20 members of labor. And then, since we are imposing more labor, it is high labor cost also. And major problem in this is loss of money due to over ripening. If in case we did not harvest at proper time, there will be chance of over ripening of the fruit. Because of over ripening, farmer get loss. So these are the major challenges in manual harvesting. To overcome these challenges, if we develop any machine which can do n number of harvestings and also which can replace the huge labor and then which can reduce the labor cost also and which can harvest at proper time, it will be more beneficial here. This is my concept model of product innovation. There are majorly 6 major components. Those are, the first thing is recognition unit. The recognition unit consists of a camera which has to identify the matured capsicum as like a human. So we should give training to the camera in the field, in the real-time situation, it has to identify the matured capsicum. And the second thing is gripping unit. There will be a servo gripper which can grip the capsicum which was identified by the recognition unit. And the third one is cutter unit. This cutter unit will cut the peduncle of capsicum. And then for mounting all these 3 subsystems like recognition unit, grasping unit and cutting unit, we require a robotic arm which can operate as like a human arm for plucking the matured capsicum from the plant. And we require a moving platform for mounting the robotic arm, and finally a moving system. For my research work, I have considered 2 varieties of capsicum, that is red color capsicum and yellow color capsicum. I haven't considered green color capsicum. And if we see the color change in different stages, in the initial stage, it will be in green color only. In the pre-harvesting stage, it will turn into brownish color, and then in the final stage, it will turn into complete red color. In case of yellow color capsicum, in the initial stage, again, it will be in green color only. It will turn into yellowish green, and then it will turn into yellow color. So, I have classified 5 classes based on the color, that is green, brown, red, and then greenish yellow and yellow. Based on, I have given a set of data training for identifying the matured capsicum. Here my motto is not to harvest the green color capsicum. Let's see the sequence of operations here. First we should identify the matured capsicum by using depth camera, and then the gripping unit will hold the matured capsicum, and then the cutting unit detaches the matured capsicum, and then the robotic arm drops the detached capsicum in the collecting system. These are the sequence of operations. I just want to show a small work which I have done. I have given a set of data of different capsicums at different stages. And in the output, it is showing like greenish yellow, it is ready to harvest. So this is the laboratory image processing method. Now by using this algorithm, I am going to do in real-time situations. If you see here, green color it is showing not ready to harvest. Impact of this robotic harvester. It will reduce the labor cost in reply to harvestings, and then it will reduce the drudgery and early detection of the matured capsicum. This will overcome the over-ripening problem also, and harvesting at right time, minimizing the financial burden and assure the quality product. Scope and utility for industries. According to World Economic Forum, by 2025, human performance will drop to just 48%, and machines will perform remaining 58%. This can state like how the world is growing fast to machines than working as humans. And Government of India, Niti Aayog's artificial intelligence strategy identifies 5 basic sectors in which artificial intelligence is more important. In this, agriculture is one among that. And as my colleague said, based on FAO reports, agriculture labor are decreasing. There is significant decrease in agricultural labor and increase in demand of capsicum production, and also increasing the labor cost. These factors compromise to say the importance of this robotic harvester. And there are government subsidies also for protected cultivation of capsicums and high valued vegetables. This is the scope and utility of my innovation. Thank you one and all.
Thank you, Ravori. Though there are a lot of paper rolls or market rolled out, what's new in novel technique that we see? We are inviting Mr. M. Jairaj, Department of Agronomy, to talk on developing novel establishment technique using mechanical paper roll for direct seeded rice.
Good evening to all, one and all present here. Myself, M. Jairaj, doing second PhD in agronomy under the guidance of Dr. R. Durai Singh, Professor and Head, Department of Agronomy, ACNRI, Madurai. Our innovation topic is on developing novel establishment technique using mechanical paper roll for direct seeded rice. Coming to scenario, rice is the major food crop all around the world and also in India. Due to uncertainty of rainfall in the command areas, water release was delayed due to the delayed onset of monsoon and also water availability in the dams. So, coming to food security, rice is the major crop. So, we have to ensure the food production. For that, direct seeded rice is the best option to enhance the food production. In direct seeded rice, we are following always broadcasting and drum seeded rice. In that, some uncertainties are there. Overcoming this issue, we developing an economically viable product, and also this innovation is an disruptive type of innovation. What are all the challenges in conventional method? Now farmers are following broadcasting and drum seeding method of direct seeded rice. In this method does not offer the proper line sowing option, and scattering of seeds in the drum seed are also a major problem. Due to this issue, and seed rate is also high. In drum seeder, up to 30 kg needed. For broadcasting, 60 kg needed. But in our method, only 5 to 8 kg seed is recommended for 1 acre. There is a high need of labor and also water requirement. This is the components of our innovation. First, using the biodegradable paper roll, we are winding the seeds along with biofertilizers and biocontrol agents. Using the mechanical paper roll paddy seeding machine, we are inside the paper roll, we pasted the seed along with thread for ensuring the stiffness while applying into the field, we can embed the seed with the paper roll directly in the machine, and we take into the field for sowing. This will give the better seedling establishment, and that will ensure the higher tiller production and also results in better yield. What is the impact of innovation? Seeds are embedded with the paper roll. It will gives the identical placing of seeds in equal proportion and equal spacing. It will be effective for the small landholding farmers below having a landholding of 0.5 hectare. Even the farmer can also bring the machine and he can make the paper roll and he will place it in the field. It will offers the precise line sowing like SRI method. It will ensure the using of conoweeder for weeding, because weed management is the major problem in DSR method. This will alleviate this issue. What is the scope and utility for the industry is? Industry can work with the student in a project mode. Industry can develop the machine and it will commercialize the machine for the farmer use. It will create a new business opportunity to the industries. This will be great demand in command areas. We worked for the Periyar Vaigai command area, and it will be suitable for all over India and Tamil Nadu, like Kaveri Delta region. Already Punjab government and Haryana government announced a subsidy for direct seeded rice per acre 1,500 rupees due to the water conservation issues, because compared to conventional method of rice cultivation, more than 15% water availability will be lesser for water usage will be lesser for direct seeded rice. It will be create a national level outreach for the this technology. If an industry can uptake this innovation as an new technology. Thank you.
Thank you. The last batsman of today in the presentation, Reem is Komanabai Raju Yadav, Department of Farm Machinery and Power Engineering, Coimbatore, to talk on design and development of automatic inter and intra-row weeder. It's an incremental innovation.
Thank you, sir. Thank you, sir. Good evening to respected sirs, madams, and my dear friends. I have done my research in design and development of inter and intra-row weeder under the guidance of Dr. A. Surendra Kumar, sir. We know that weeds are the unwanted plants which grows in the crop. So, those are create harmful effects in the crop. In developed countries, we know that 5 to 10% of weeds are there and crop yields damaged. In India, more than 50% are there. Here we see that in this picture, the 45% of weeds are affected the crop yield, and in this reduction of the crop yield, not only the reduction of the crop yield but also the crop quality, and due to this one we are increasing the crop cultivation also, and weeds are more competitive for the nutrients, water and sunlight. So according to the Indian statistics, chili production is the first place, and next one is tomato is the second place. We will go for first cultivation area. In tomato, 7,000-9,000 hectares are cultivated, and after that chili 3,09,000 hectares are cultivation. Production, you see that the production is more in tomato, that 19,759,000 metric tons, and chili 3,592,000 metric tons. So that means you see that the production is more in tomato and chili also here. And also daily wages, daily the farmers, and not only farmers, everyone using tomato and chili. Production yield reduced by the percentage. Here you see that tomato 53 to 67% due to the weeds. In chili 60 to 70%. That means the yield quality and also the crop production is reduced by the weeds. So before I am going to explain about my concept, what are the existing practices are there. Firstly, we are seeing that manual practicing. The weeding activity is a strenuous one, and which is a tedious work and labor-intensive process. Mainly, the women are adopting squatting and bending posture, which leads to increase the heart rate. You know that heart rate increases because they are doing continuously weeding per day 30 to 240 minutes, and also they have done weeding in particularly season about 15 days. So they also got musculoskeletal problems. And also the labor intensive process. Here you see that 86 hours per hectare required for manual laboring. So drudgery is reduced by self-propelled weeders, but self-propelled weeders cover less area only. Here you see that 0.04 to 0.05 hectares only, but they cannot do in the intra-row weeding. You know that intra-row weeding means in between plants they cannot cover the weeding completely. Here you see that self-propelled weeder can cover in only one row, and when compared with the self-propelled weeders, tractor-operated inter-row weeders, you are seeing in this display here that it can cover the area about 5 rows at a time. So the width of the operation is more, so we can save the cost like 52%, and also the time 78%. Here we see that it requires only 22 hours per hectare. After that, updated one is that herbicide applicators. Herbicide applicators mainly shows negative impact on who sprays on the field. Why? Because they are not following the masks, helmets, those things, and they are following the herbicides and post-herbicides, which leads to reduce the field quality. And also nowadays, a lot of people who educated and who knows the knowledge about, they are encouraging the natural farming to eat the better food and better quality of food. Here we see that in picture. So to overcome these challenges, I have taken this inter and intra-row weeder. Before I have designed the inter and intra-row weeder, I have done a small research here. You see that the first picture, the weeds I have taken after the sowing of tomato and chili at 15 days to 45 days. I have taken the data. Root growth of the weeds doubles than the crop growth. That means which can compete for the water, nutrients, and also sunlight. And I measured the weed population, 1 by 1 meter square population, how much weeds are there separately in inter and intra-row weeder. I have calculated here. In this figure, you see in this tomato and chili crops are there. In that around 3 distinct types of radius are there. In this distinct type of radius, why I have selected means I want to rotate the active tool, nothing but which can rotate around the plant and remove the weed. I have taken and I have measured the weed population. After that, final dry matter content. That means how much losses occurred due to the weeds I have measured. Not only the dry matter content, but also I measured the moisture content of the weeds. So why? Because how much water absorbed by the weeds, that means which can give the compete for the crop growth. So from that, what are the advantages here? I already mentioned before that it reduces the drudgery and saves the time and cost, reduce the usage of herbicide applications, efficacy of our inter and intra-row weed is more, and finally AI reduces the human interventions. So that means we can reduce the human labors and also inter-rows which can require more time. So this is my final conceptual design here. That first you see power from PTO. We can connect the tractor PTO to our weeding tool by using universal joint. The weeding tool you see in that picture, that is the active tool which can rotate. The trajectory motion of the weeding tool is in cyclic or triclic. That means which can rotate in circular. After that it can go straight. So that is the active tool. You see in that duck food seeds. Those are the passive tools. So that means that can be maintained linearly. We can give the depth gauges. So duck food seeds can run in inter-row only. By using this, we can synchronize the forward speed of the tractor with the weeding tool. then only we cannot damage the plant. After that only we will go for artificial intelligence, which can reduce the human drudgery and saves the time, and which improve the efficacy of our inter and intra-row weed. So impact of the innovation. By using the sensors, automations, we can do the weeding, and also effective utilization of power. So that means we can use effectively and efficiently by using our inter and intra-row weeder. By using these things, we can increase the productivity. Finally, we can go for the product of inter and intra-row weeder. Thank you.
Thank you, presenters. Okay. Okay. So quick questions, rapid questions. Be ready. All of you come to the front now. Against the backdrop of the backdrop. Question and answer session from the audience. This time is up. Validation is going to begin. Make it fast, no? Any questions you can ask. We are readying for the valediction. Vice Chancellor is coming on the way.
Check. This question is for Surendra Kumar. Your inter and intra-row wheeler concept. It's here.
Me, sir. It's me, sir. Inter and intra.
Okay. The inter and intra-row weeder, right?
Yes, sir.
So you had a picture or a framework of that row weeder.
Yes, sir.
Which has to be hooked onto a tractor, correct?
Yes, sir. Tractor only, sir.
Is it not possible to have 4 wheels for that and a remote control unit so that you don't need a tractor?
Sir, in Indian conditions, the precision of row, that means deviation of the sowing, transplanting occurs, sir. That's why only we are choosing the tractor, sir.
I'm actually asking you that question only from an IoT perspective, yes, because any hindrances on the row,
yes,
and any blockages or turns,
yes,
a machine learning algorithm will be able to build it in, so that what will happen is you will have an advantage of you don't need a labor at all.
Yes, sir.
So maybe you should think on those lines.
Okay, sir. Okay, sir.
See, it's just a question of attaching a remote control unit to it.
Okay, sir.
And a sensor at the front which looks for blockages and hindrances.
Okay, sir.
So there,
sure, sir,
further cost saving is even the driver or the tractor and the fuel, those costs will also be saved.
Sure, sir, definitely, sir.
So this is an actually advancement he is suggesting. This is what actually the convergence happens. And you have developed a weeder which has to be mounted with a tractor. Now I think if you put some sensors into that one, and if it can operate by itself, then I think it is more easy. There even for a tractor you need a labor, but without a labor you can achieve the same thing. So probably an advancement you can think in the later stage. Okay, it's a good suggestion. Yeah.
I think the same thing is also applicable for Mr. Jayaraj, who did the mechanical paper roll. So
Yes,
you
sir.
also have a mechanical device which is pulled by a farmer in that photograph, correct?
Yes, sir.
All you need is a remote control unit with those sensors and go on and paths, much more cost savings, and it's an independent operation.
Okay, sir.
Think about it.
Okay, sir. Okay, sir.
Any more questions from students? Yeah, come.
I'm Gokula Krishnan from 2nd MSc Biotechnology. I have an issue and doubt regarding the spray with drones. Whenever we have a spray there, we have a face a problem of drift, spray drift to winds. Actually, as drones also wind propelled, won't there be any drift in the spray?
Actually, drift is a major problem, but we have to take precautions during applications, and the wind speed less than 6 km per hour, so we can apply the herbicides or any other chemicals. So the drift will be minimum.
You're saying that the propulsion from the drone won't be affecting the sprays?
No.
Thank you.
Hello, Raju Yadav. Sorry, Rauri Shai Prashant. My question is, your robotic harvester comes under, I think it's machine learning, right?
Yes, sir.
Okay. Which kind of algorithm did you use? For example, supervised or unsupervised?
Supervised classification only,
Okay.
sir.
Then என்னோட question என்ன அப்படின்னா, for example, in the harvester testing period, four types of capsicums நான் வச்சிடுறேன். Same time, artificially create பண்ண 4 different colors of capsicum வச்சிடுறேன். So that அந்த robotic harvester வந்துட்டு எல்லா capsicum-ஐயும் pick பண்ணுமா இல்ல original capsicum மட்டும் pick பண்ணுமா?
Actually, camera will be having a depth sensor, sir. So which one will be forwarded, I mean, which is ready to harvest. They cannot be a single line, sir. So they can be at a place, but one after one or like that.
Okay.
So
then,
based on the distance from the camera to the capsicum which is matured, it will sense the first one and will detect, I mean, which will harvest the first one, sir.
Okay, நீங்கள் சொல்கிற அந்த கேமரா அப்படிங்கிறது சென்சார், ரைட்? இப்போ நம்ம சொல்கிற போகிற அந்த கேமரா எல்லாமே சென்சார். அந்த சென்சாருக்கு வந்துட்டு அதாவது ஆர்டிஃபிஷியல் கேப்சிகம்னு எப்படி தெரியும், இது வந்து ஒரிஜினல் கேப்சிகம் அப்படிங்கிறது எப்படி தெரியும்?
சார் அது தெரியாது சார்.
தெரியாது. ஓகே. பட் அதோட எக்ஸ்ட்ராக்ட் ஃபீச்சரை நீங்கள் எக்ஸ்ட்ராக்ட் பண்ணும்போது அதால் ஐடென்டிஃபை பண்ண முடியும். ஸோ தட் ட்ரை பண்ணி பாருங்கள். ஓகே. ஒரிஜினல் கேப்சிகம்க்கு சர்டன் ஃபீச்சர்ஸ் இருக்குது, ஆர்டிஃபிஷியல் கேப்சிகம்க்கு சர்டன் ஃபீச்சர்ஸ் இருக்குது. அதை நீங்கள் எக்ஸ்ட்ராக்ட் பண்ணும்போது உங்களோட ரோபோட்டிக் ஹார்வெஸ்டர் கரெக்டான ஆப்ஜெக்டை மட்டும் பிக் பண்ணி எடுக்க சான்சஸ் அதிகமாக இருக்குது.
ஷூர் சார்.
ஓகே.
தேங்க்யூ சார்.
Sir, I have a question for Mr. Ravi Rao. Yes, for the capsicum one.
Yeah.
Yeah. So, in your model, will it detect whether there is a pest borer or not?
What is it?
A pest. When capsicum fruit has a particular borer in it, when you pick it up, won't it ruin the good produce also?
Yeah, actually, the program algorithm is only for identifying the maturity or not. It is not for pest and all.
Then the efficiency of it, isn't it questionable?
What efficiency? Like, even it is pest affected, if it is matured means we have to harvest.
Okay, thank you.
Hey, hi, I have a question for Jairaj. Jairaj, you said your seeds will be placed on papers, right? So what kind of papers are they?
It is an paper roll which is biodegradable, which is used for putting in a table for having food now. That paper is only we are using.
So how many days it will take for the paper to degrade in soil?
It will take 25 to 30 days for degrading in soil.
Okay, in that case, don't you think the seed will get any kind of fungal attack within the time of its germination then?
No. Within 3 days, first 3 or 4 days, germination will emerge, seedling will emerge. If you came to stall means you will see in the, I put a demo for germination in the paper roll seeding.
In case of when the paper is kept within the soil, I think your concept is that, right? You'll be keeping the paper, the seed will be placed inside the paper and the whole thing will be placed inside the soil. So in that case, within that time there will be no light for the seed too. So
which it was not placed inside the soil. It will be, field will be properly leveled, puddled and leveled before on the leveled soil. Above portion only we have placing, not placed inside the soil.
So in case of rice you are speaking about?
Yeah, only rice.
So don't you think the paper will imbibe the water and it will immerse?
That paper will absorb only water, not imbibe or immerse in the soil.
Okay, fine.
I do have a same question
The
on
session
that.
comes to closure. Now the valediction will be beginning Then a short while.
Sir, same the seed packaging, he said, no, sir. Is there a chance of packaging the seed protectant materials like fungicides or growth promoting materials?
I already told, we are mixing the biofertilizers as coating. We have also coating the biocontrol agents, fungicides, all the growth regulators we can add it in the seed paper roll.
Okay, thank you.
Thank you. The validation will begin within a short while.
So, thank you. So, all the technical session is almost closed. Please remain seated, and our Honorable Vice Chancellor is just will be arriving in couple of minutes. Then we will start the validatory function. Probably we'll finish it in another 30 minutes. So please be seated and for this validatory function, and almost Vice Chancellor is arriving very shortly. Thank you.
Good evening, ladies and gentlemen. It's a pleasant evening to one and all present here. The valediction and ado ceremony begins now for the two-day global conference on Expo on Agro Innovations, 19th and 20th July. Now, before going for the main program, we got Hosanna, praising the Lord. May I request the delegates on the dais and off the dais to upstand for the prayer. Thank you. We are delighted to invite Dr. A.R. Lakshmanan, the Dean, School of Postgraduate Studies, to offer the welcome address and to relive the moments for the past couple of days.
So, good evening, everyone. I feel very happy to welcome our Honorable, most respected Vice Chancellor for this valedictory function of the global conference. Welcome you, Madam. I think you participated in the inaugural session and gave a lot of motivation and also gave good suggestions. And these 2 days, I think it went really well. I think personally, I am very satisfied. And that's the way, this is not just I am telling from the bottom of my heart. This is a maiden attempt, you know that, done. And personally, I think I am extremely satisfied. So that is the first commitment I am giving to you, that we are pretty happy with the proceedings happened. So I welcome you, Madam, for this valedictory function. Then I have to inform that Dr. U.S. Avasthi, Managing Director, IFCO, New Delhi, and Managing Director, Sir, will be joining in the online very shortly. And you know that IFCO is doing a great service to Indian agriculture, and by the way of producing major nutrients, urea and phosphate and potash. And very recently they launched nano urea. IFCO launched nano urea, which has been a big success. And you all know that IFCO has been associating with our university for the past 3 years through various projects. And our Honorable MD of IFCO will be joining, because just he has landed Delhi airport, and he said that he'll be joining very shortly. So I think on behalf of the entire university, and we welcome our Honorable Managing Director of IFCO, who will be addressing these students very shortly, as soon as we get his line through, and he'll be talking with us over through online. So I welcome Dr. U.S. Avasthi, Managing Director IFCO, for this valedictory function. Then I take great pride in welcoming Mr. S. Jayaraj, Associate Vice President, Bull Agro Implements, Coimbatore, and so who are involved in the manufacture of agro implements for quite some time. So we welcome you, sir. And I take the opportunity to welcome Mr. Jay Prashant. And I think he just before he gave on lead speaker talk in the AA session, but today I think he is representing CAA, because we all know that CAA has been part of this 2 days event, and he is also a part of CA. So I think today he is representing CAA to extend his feedbacks and as well as vote of thanks to the audience. So then I take the opportunity to welcome all these industrial people who have joined through actual and as well as the virtual mode, and all the directors and deans of Tamil Nadu Agricultural University, heads of department, and press people, and my dear student friends. So I welcome you all, everyone, for this valedictory function. Now, just before I leave the stage to the speakers in the dais, I would like to relive the moment. That's what the topic given to me. What has happened for the past 2 days. So sometimes, before, I think we have prepared one small audio visual. Sometimes pictures can show things better way. Wait. So go to the previous slide. Yeah. If you're ready with the audio, no problem. Yeah. Audio is ready? Yeah. Okay. Then we have prepared one audio which will help you to relive what happened for the past 2 days. Yeah. Thank you.
and I came here. It was very nice. And the people from Madurai and Coimbatore, all the PhD students are very, very talented. And the one thing which I observed that they are actually seeing the pain point, and then from the pain point they are developing their project, you know.
Yeah, I have gone through this Global Conference Students Innovation Expo. It's well organized. Students exhibited their research work in a very applicable manner, and it's a very good platform for the students as well as for the industrialists to get contact each other and also to move further for next level.
இப்போ வந்து it was very useful for us. ஸோ லைக் நிறைய செடியெல்லாம் வளர்த்துறேன், எனக்கு இதில் கொஞ்சம் இன்ட்ரெஸ்ட் இருக்குது. ஸோ அந்த மாதிரி இருக்கும்போது இட்ஸ் வெரி யூஸ்ஃபுல் ஃபார் மீ. ஸோ இது எப்படி ஒரு நார்மல் க்ரோத்தை விட சில இதில் இது பண்ணும்போது அது ரேண்டமாக ஃபாஸ்ட்டாக எப்படி க்ரோத் ஆகுது. ஸோ அதில் எக்ஸ்ப்ளனேஷன் எல்லாமே சூப்பராக தந்தாங்க. ஸோ இட்ஸ் வெரி யூஸ்ஃபுல் ஃபார் மீ.
It is the overall view of this expo and your conference is really good because it gives an answer and a solution to the current scenario. You know very well that today it's a big problem with the fertilizers application, and there is a big demand for the fertilizers, chemical fertilizers. It is not available in the global scenario, not only in India. The expo, some of your stalls which has given a solution for this kind of a thing because it is more on the nanotechnology. So there is a good demand and good opportunity for the nanotechnology products, and also you have some of the unique products which your people have invented.
Slide. Yeah, of course. Thank you very much, Sridhar and Prabhu and Ganesh and every team. Just in a very short period of time that they prepared this particular video, just which really helped us to go back what has happened. Now it's business time. So I have to answer our Honorable Vice Chancellor, what is the real outcome of this particular meet. So whether we have achieved what we thought at the start of the meeting. Have we made a real connect with the industries? Have we exposed the students to this industrial platform? So probably I think I have one slide. So almost close to 17 companies have already given their consent to work with TNAU. So that is the biggest, I say that it's a success for us, and they have in principle have agreed, and just I am giving some information to you. So National Agro Federation interested in coal plasma technology, seed drilling technology, nanopellets to take it next level. Linga Chemicals interested in humic acid based formulation and chelated micronutrients. Then Cryogen Biotech and AgriApp, and they are interested in multi innovations, and they are interested to come down separately for a discussion because being a corporate company, they want to take it in a more orderly way. And Zero Harm Sciences Private Limited, Hyderabad, they are also interested in phytopharma. And IFCO MD sir has already promised that whatever the help we ask for industrial internship or joint research, they are ready to be on board with us, because already they are on board. They want to further strengthen their collaboration. Then BG Crop Science and Technologies Private Limited, they are much interested in nanonutrient. At this point of time, I have to mention that BG Crop Science has they are magnanimous in providing a special prize to all the innovators, which is worth of close to 1 lakh rupees, and which is going to be distributed by our Vice Chancellor today. And that particular, probably, I don't know what that particular memento contains, but I think it's going to be a good, what I'm saying is gesture by the BG Crop Science, because the BG Crop Science chairman, he observed some of the presentation, and he said that we have to motivate the students for the confidence they are showing. So that is the BG Crop Science, apart from the MOUs. Then Harrison Malayalam Limited, I think they are interested because they are also associating with nanotechnology for TMB management. Now they want to enter into tea nutrition with MOU. Then CABI, and Madam knows it pretty well, and CABI has already given consent for industrial internship. Yesterday, after the discussion with Mr. Gobi Ramasamy, many students met Gobi Ramasamy in person, and they expressed their willingness. And Mr. Gobi from CABI, and he mentioned that personally, please continue to work with us and follow up with us, so that I think immediately whatever can be done, we will initiate. From this year onwards, they are ready to take for student internship for a period of 3 months to 6 months. Only thing is we have to formalize those things. That is in the case of CABI. And today morning, WRI, Mr. Nambi, Dr. Nambi, he mentioned that, and after seeing Mr. Gobi Ramasami's interest, he said that they are also ready for, but they, WRI is bit specific. They want to collaborate with Agricultural Economics Department. And you know that WRI is mainly working on policy level matters. So he said that he is ready to work with our own students for internship for 3 to 6 months, mainly for agricultural economic students who can be trained by them in policy, drafting policy documents like that, which is really a good thing to happen. Then Core Garments Private Limited, again I talked with Dr. Mr. Niroj Mohandi. He was mentioning that anyway that because already they are working on converting the various carbon services into carbon trading platform, and he said that he's again come down to here to sign a specific MOU with the TNAU to convert the eco-friendly technologies, to promote eco-friendly technologies into carbon trading platform. So that is in the case of Core Carbon, and Trinita Technologies are already there, and they are ready to work with us on IoT and AI, and they are ready to train 25 students free of cost to train on IoT and as well as AI. And this is in the vision of our Honorable Vice Chancellor, that is developing a school of finishing and grooming. And the first milestone is 25 students from this year after they finish their post-graduation, either MSc or PhD. Now the ball is in our court. We have to come out with a program so that I think they can train the 25 students. Not only training 25 students, they are ready to absorb the 25 students for their company. So this is another thing, and probably this Prashant is there, and Mr. Rajan is there. We have a discussion, anything. And in the case of last TVIA, they are interested in any nano encapsulation of functional molecule. And finally, Coromandel people also interested on crop nutrition. So these are some of the things initially happened. I mentioned this is just a starting point. Probably things will evolve when we start working without losing the momentum. That's what actually I request all the heads of department directors. This is an initiative done by the Dean SPGS, of course, not Dean, it's by the School of Postgraduate Studies. It's just an initiation, but it's a teamwork. At the end of the day, almost all these innovations have come from the different departments, from different directorates. And how we are going to take it forward without losing the momentum is very, very important. So I request everyone, because the innovation abstract is there, companies interested, all these things will be circulating very shortly. Now it is up to the concerned heads of departments and the directorates to take it forward. But we will create a post-conference desk. As we promised, we'll create a post-conference desk. We'll circulate the information of all the companies to you. We can only facilitate. But I think I request that the concerned people should take it forward. So with this, then a couple of more information about this conference. Almost as I mentioned, 6 themes we conducted the conference, 1,600 participants registered, and in person 900 participants, and then close to 700 participants online participated, and more than 800 members visited this conference in 2 days. This is from our own scientists, students from Kumaraguru Institute of Agriculture, Erode, Vanavarayar Institute of Agriculture, Pollachi, likewise many colleges, then Akshayam International School, Coimbatore, industrialists, entrepreneurs, our own SRFs, JRFs, and it's a assorted group of people visited this one. And then of course, this created a good platform for the students to present their innovation in the presence of various industrialists, and a lot of cross-learning happened through this process. So this is a small summary of what has happened in these 2 days. I personally feel that the MD IFCO is online, please. already joined? Okay, then then sir can speak. Yeah, we can. Okay, okay. Thank you, yeah. Yeah, I think I can see, sir. Okay, right, yeah. Thank you, thank you. Yeah. Sir, we can see you, sir. Welcome to this conference. You can speak, sir. Thank you, sir. Hello, sir can speak. We can hear him. We can see him. Yeah, yeah, yeah. Unmute. You got to unmute him. Yeah. Wait, wait. So you please unmute yourself. You have to unmute. You have to unmute. Yeah. No, you have to unmute. Sir has to unmute. Yeah. Being unmute, but I think we have already unmuted. So, அங்க unmute ஆகாம இருக்குது யாருடைய சைடு இது பாருங்க unmute ஆகாம இருக்கு. Yeah, one minute, we are unmuting. Yeah, yeah. One minute. Yeah. Now sir can speak. Yeah. Yeah. Yeah, just let him speak. Let me try whether his voice is clear. Let him speak. Yeah. Still, he has to unmute because we can't hear his voice, but we have already unmuted.
Hello.
Yeah, we can hear.
Can you hear, please?
Yeah. Yes, sir, we can hear you. Yes, we can hear you very much, sir. I will introduce you, sir. 1 minute.
I can't hear you.
Sir, we can hear you very well, sir. You can speak, sir. We can hear you.
Oh, thank you very much. My dear honorable speakers on the dais, my friends from the student community, respected professors, ladies and gentlemen, I have pleasure to participate in your valedictory function, and it has given me immense satisfaction to be part of a group which is going to look after the future agriculture. We all know that agriculture is the oldest industry in the world, and it has gone through a numerous innovation in agriculture. Depending upon the type of soil, the type of weather, over the centuries, different kind of crops have been grown in different kind of soil, and depending upon the climate, they have nurtured to the level that they found market either in their own country or in those countries where, where there was a requirement of that product. Out of agriculture, lot of our industry, other industries have been evolved. Like, dairy industry, sheep breeding, horse breeding, camel breeding, and that is where the domestication of the animal power started. Then came, as and as population increased, the necessity of providing plant nutrition came. And then came the chemical fertilizers. Then after that, with agriculture becoming more intense and the effect of pests, which started destroying the crops, then farmers found pesticides which could take care of those pests. But that has deteriorated our soil content and soil quantity and soil quality. So now the time in this 21st century, we have to think about agriculture the requirement of plant nutrition became very important. And as the science evolved, my dear friends, science evolved very rapidly into nanotechnologies, into space science, which had a very major role in nanotechnology, IT industry by way of chip had the role of nanotechnologies, and medical research has a lot of role into that. But agriculture was getting deprived of that research. So IFCO, which is owned by the cooperative of the farmers, did a pioneering research work of bringing nano fertilizer into play in our country, in India. It's the first time that we have invented nano urea, where 20 gram in a 500 cc bottle is encapsulates the nanoparticle of nitrogen, and they are then released by way of spray. And that is where, what happens is the interest in having more nano kind of fertilizers came into play. We are into very advanced stage of doing research, and we are in very soon we will be also in the field with nano DAP. Already, as far as the nano urea is concerned, we are already building a lot of plants. You all, dear friends, you all know I think, dear friend, we all have our role and we all need to be together, industry, academic, and right, and the students like you should be
சார்
there
அப்படியே இந்த
to
லைனை
help
கண்ட்ரோல்
us
பண்ணுங்கள் சார்.
in achieving that.
சார் கேட்குறேன்.
நீங்கள் டோன்ட் டிஸ்கனெக்ட். உங்கள் ஸ்பீக்கர்லேருந்து எனக்கு கேட்குறது, நீங்கள் டோன்ட் டிஸ்கனெக்ட்.
Yeah. Just a second. No, just a second. From your end disconnected.
No. You please speak because I am connecting through your mobile phone. Please connect. Continue. We can hear you.
Yes, sir.
Don't disconnect this call, please.
Not disconnected. Yes, sir. Acha. This call you are joining now.
Yeah, let him speak.
On this call.
Yeah. No, we can hear whatever he speaks. No problem. Please carry on.
Hello.
I can hear you, sir. Please carry on.
I can carry on.
Please carry on, sir. We can hear you. Please.
Okay, thank you. So I was saying, dear friends, what we need to do is that we have to form a kind of a club, kind of a mechanism by which a startup by young students, young scientists, and together with the help of guidance from the seniors of the institute, we in Expo will like to support your ventures, and we invite you to help us in making these nano products more efficient, more farmer friendly, and ultimately our dream is that nanopesticides and nanonutrients together should be able to take care of chemicals from the soil, which will reduce our dependence on fossil fuel and make our country very strong. As our Prime Minister said, Atmanirbhar Bharat and Atmanirbhar Krishi, the self-reliant India and self-reliant agriculture is the need of the hour. And today's world, when we had corona and other disturbances in the world, there's more requirement that we should do our best to make our country strong, and we should make our country strong, and we should feel proud of our country. With these few words, I would like to thank you for listening to me, and God bless you, and stay and have good health. Thank you very much.
Sir, thank you very much, sir. I think in spite of your hectic schedule, I know that you are returning back from the airport, and in spite of that hectic schedule, I think you managed to squeeze time and talk with our students. It's really inspiring. Thank you very much, sir. Thank you, sir.
Thank you. Thank you very much. Thank you.
Thank you, sir. So now I request Mr. S. Jayaraj, Associate Vice President, Bull Agro Implements, just to say a few words about this particular conference.
So good evening, everyone. First of all, I would like to thank the Honorable Vice Chancellor and Dr. Lakshmanan for giving me this opportunity. I have immense pleasure in having a view of your exhibits, and it's very good actually. And I think we are marching towards more innovations. I represent from Bull Agro Implements. So it's a company we started in '98, where we do the tillage equipments and blades and tractor drawn implements. So we have been in this business for more than 25 years now. So one few things I would like to quote, like one, now the fields are shrinking. So whatever the innovative products we are developing, it should be with the farmer friendly and affordable to the farmers, and the performance should also to the expectations of the farmers. So these three things I think we should keep in mind and do the products. Two, I think we have to have close interaction with the industries where we can be a bridge to the farmers. So one side you can innovate the products, the other side we can carry to the farmers. So we will be acting as a bridge. So we are open to support. And I would like to take this opportunity to tell you that we are open for internships and projects, also final year projects, which we can give it. So with these words, I wish you all the best. Thank you.
Imitation is the best form of admiration, so also feedback is the best form of assessment. We are inviting 2 students volunteers who can come out with the feedback on this 2 days conference. Any 2 can come and join very shortly, please.
Good evening, one and all. First, I would like to thank our Honorable Vice Chancellor Ma'am and our Dean Sir for encouraging us in presenting the innovations before the industries. This is a greater opportunity given to us. We as a students, we can work, but the guidance to promote our innovations is very important. And now we have got this platform, and all the people involved in conducting this program in a successful way. I thank all the people, and I also happy to see my fellow friends in a forum, so that we could know what are the innovations going within the university itself. Because though I'm from nanotechnology department, but still we were not aware of other innovations going on in our own university. But we are very happy to see all our fellow friends from our other departments also, and now we are having ideas how to link innovations, or how to create a link between different interdisciplinary also. So I've got n number of contacts within students and industries also. Thank you, sir. Thank you, ma'am, for creating this wonderful opportunity. Thank you.
Yet another one can join.
Good evening to all most respected VC, Dean, and all professors who are here. Myself, BS Agri. I am running a proprietary company. Really, this conference will be very helpful for us, madam. What to do, how to move with industries, and really we have more contacts. And these are the things that are available in the market. We are able to know, madam. Also, one thing I want to ask you. We are doing nowadays. Everybody want to do organic farming. Madam, question. Nowadays, everybody needs organic food, madam. I am doing organic farming in our native place, Rajapalayam. 10 acres, we are doing organic farming, madam. I need to do research on organic farming, madam. Like in chemical farming, for urea, this much quantity is given to the plant. But in organic farming, there is no such specifications. Even though we are doing, madam, we got amazing results. We have 5 acres of Taiwan pink guava. we are getting good results, aroma, taste, everything is good. But I want to do it legally along with the university, for which I need your support and help, madam.
Great. Thanks for coming forward to join with the university and take your career up. And university will definitely be supportive of. We have got separate Nammalvar Organic Research Center. I will request the scientists to be in touch with you so that you can, we are standardizing many things in the organic farming, especially how much to apply, what quantity is required. Just like chemical farming, we are also developing standard operating protocols in the organic farming as well. We will definitely would like to work with you, and it will definitely help both you as well as the Tamil Nadu Agricultural University in refining certain important areas. Thank you so much for coming forward.
It's my pleasure, madam. Thank you.
It's scintilla time for the distribution of the certificates for the innovators. Let me know. One more volunteer.
Thank you, VC ma'am and Lakshmanan. I think it was a fantastic eye-opening session for me personally. I come from a company called Green Collar Agri-Tech Solutions. We felt that both the white collars and the blue collars are supported by green collars. So we need to now bring dignity to green collars, and we need to do something for that, and that is why I'm here. We are incubated at TBI in our own TNAU. And it was such a eye-opening session, and during the lunch session, I interacted with a lot of PhD students, and I found that everybody unanimously told me that this is the first time we are doing, and all of them uniformly told me only one thing, that we have gotten our confidence in this session, which was very, very good. So I think you should all give a loud applause for yourself, and it is such a, for me personally, it is such a pleasure to see a women leader who's kind of here. I think after 12 years, we've got a women leader. And I can see from every action of our VC, I would not call your VC, our VC, I think that she'll be a pride of Coimbatore, and we are all here to support her and the initiatives. And ma'am, I want to tell you that after you left, all the sessions were very interactive. They asked so many questions, and the fear is gone. So I think this is the first step. More to come, and then I hope to mentor many entrepreneurs. I'm coming from a commercial industry, so much guidance can be given. So wish you all the very best. Thank you so much.
Could we request our Honorable Vice Chancellor to offer the valedictory note?
Good evening to all of you. The most respected Dr. U.S. Aswati, Managing Director, IFCO, New Delhi. You know, he's such a busy person, such a great personality, and he was kind enough to share his thoughts with us. Unfortunately, there was a little bit of inconvenience because of the internet connectivity, but still, it is a pride that he could participate in our conference. And IFCO has done a lot of things, and now, you know, the urea, if we go with the prilled urea, the use efficiency is around 40%. They are venturing into nano urea, and nano urea, definitely the use efficiency will increase. And at that context, his presence was really important for this conference. And thank you, Lakshman, for organizing that. And the second one, Mr. Jairaj. So we have Bull Agro implements. We have a very great connect with you, and thanks for supplying few of the cono weeders. You know, we actually recommend cono weeding for the system of rice intensification, and we were finding it very difficult to have manually operated cono weeders, and Bull Agro Enterprises instruments have come forward with mechanized cono weeding system, and we have tested in our university wetland farm, and they have also provided few instruments for our own use. Thanks for that, and we will connect with you for many more innovations like that. And again, very importantly, the Jai Prashant. Jai Prashant representing CAI. So thanks for your presence here again. And Dr. Vanitha, my university officers, industry friends, more importantly, the students and the young innovators. Really, it's a pleasure for me to be here for this valedictory function. You know, like Dr. Lakshman has his unique way of organizing things. I've enjoyed working with him since 2007 onwards. You know, like I just want to have a small story with you. I was sitting in Japan as an invited professor for a short period of time, over 3 months. I was actually writing a proposal for funding support. Then I wanted it to be truly interdisciplinary, so I just thought of few people and gave mail to few people. The first person who replied back to me in half an hour was Lakshman. So I took him as my partner for that particular project, and since then I used to enjoy the organizing capacity of Lakshman. We connected with more than 2,000, 3,000 farmers in that project over a period of a decade, and he organized things so well. And this time, the organization of this particular conference, it is marvelous, and you know, it is unique way of Dr. Lakshman to organize this kind of conference, and it requires a special applause for you, Lakshman. And again, it's amazing to note that, again, I just want to say another important thing. I'm really happy to have a few feedbacks from the students, and very importantly, feedback from Hemlata. Really, it's very important. I was told that you were the person who did not miss even a single session. The whole 2 days you are here, and you are the right person to give the feedback. Thank you for your feedback, especially the positive feedback. I'm really happy, at least a confidence level has been boosted to the students. You people also can innovate, and you people can also become an entrepreneur. That is what is the whole purpose of this particular conference. And I'm really happy that there were 17 different companies who already are going to be part of TNAU. Most of them I know personally. For example, National Agro Foundation, I don't know whether they are here. They were there. Okay, so National Agro Foundation, they are doing a wonderful job connecting with the farmers, FPOs, and we will definitely closely work with you, sir, and our students will be benefited in closely associating with you. Similarly, WRI India, it is one of the very leading research organization, and they are going to take some of the economists especially, so it's very pride for our students to associate with them and work with them. Similarly, CABI, this is again an international organization with its headquarters in Geneva. We visited that. They have got such a wonderful facility, not only in Geneva, in many other places. Here they have their headquarters for the South Asia in New Delhi. Their office is there. We have visited and we have been closely working with them. And if possible, they are really working lot on plant protection aspect. So some of the students from kind of pathology or entomology, those people, they can definitely get in touch with the CABI people to get your opportunity to go abroad and do your research. And many more things. I can list all the companies are really, really good. They have come forward to work with us. And again, I'm very happy to learn that there were 900 participants who were really in offline you have participated. So online is also good, but offline has its own advantage. Face-to-face communication, learning things very effectively. So there is nothing can replace offline participation, like face-to-face participation. It's good that I'm really happy. Many people, over 900 people had an opportunity to participate in person, and another 700 people who could participate online. So it's a great conference. Anything which is more than 300, 400 itself is a good conference. Here, more than 1,500 people connecting with the industries. It's very, very important. Especially students, universities and business, they must go hand in hand. Then only we will make our students to prepare to face the real situations, real problems, and also to come in a very, they will get ready for the future scenarios. You know, the School of Finishing and Grooming, this is not my buzzword. This is the buzzword of Dr. Venkat Prabhu. Where is Prabhu? I'm not seeing him here. Yes, he was the person who was telling me this idea, School of Finishing and Grooming. As soon as I became the Vice Chancellor, I selected my team of university officers and I sensitized them with my vision of university for the next 3 years. I was just telling about the School of Finishing and Grooming. This is one step forward towards that School of Finishing and Grooming. Lakshman has organized a very wonderful conference. I'm very happy that more than 300 companies have looked at your innovations. The way you have presented, some 100 and odd innovations you have presented, it is marvelous. And the way you explained, I really did not find time yesterday afternoon and today to come and personally interact with you for each and every innovation was very good. Definitely, I think you are going to keep your innovations tomorrow as well. You said you will be keeping it for finished, huh? Okay, so it was really good, but these are all some of the things which will boost your confidence, how to exhibit things, even that one-page abstract, what to give, it's an art of writing that one-page abstract. You can write 10 pages, but bringing that into one page is really difficult. Now you have learned what to put in that one page, how to just synthesize, like 10 page you can very easily write it. You can write stories, but if the entire thing has to be synthesized in one page, that is where your skill lies. So he has given a way to develop your skill of writing a small abstract, which actually shares what are the things which importantly needs to go into that abstract. So this way, this is a learning experience, connecting with the people. This is very, very important. I'm thoroughly satisfied with the way the 2 days have gone, and I'm very happy that even at the valedictory function, I could see more than 500 people in the auditorium. That itself shows the success of your conference, Lakshman. So, hearty congratulations to each and every one of you. Do work with the post-conference desk and try to connect yourself to the companies and try to harvest the fruit which has been developed so far, and try to have some connect between you and the companies for the past 2 days. Okay, as yesterday we were talking, this is just a beginning, right? So you have to make use of the opportunity. So we have created a way. It is your interest, your enthusiasm that has to come, and we will definitely help you to achieve your goal. Okay, so thank you very much for the great opportunity given, and I congratulate each and every one of you who have participated in this conference, as well as who has tried hard to make this conference a great success under the leadership of Dr. Lakshman. Thank you very much again.
Thank you, ma'am. Now it's time for certificate distribution to the innovators. For the exhibition who have made displays in the exhibition or called to receive the certificate from our respected Vice Chancellor. First, Ashida Joy of Coimbatore campus. Archana T, East Indore, Coimbatore. Ashwida K, East Indore, Coimbatore. Balakanesh B, East Indore, Coimbatore. Chandrasekhar Y, Cheran E. AC Indore, Coimbatore. Chinnadurai S. Chinnamani I. AC Indore, Madurai. Claudia KL. AE Indore, TNA Coimbatore. Davidson Dawkins, AC Indore, TNA Coimbatore. Gautam Golla, AC Indore, Coimbatore. Gurvi Vaibhav Ramesh, Horticulture College and Research Institute, Coimbatore. Jagdishwaran P. AC Indore, Coimbatore. Karpi Omkar Shivaji, Agriculture Engineering College and Research Institute, TNA Coimbatore. Karthikeyan G, AC Indore, Coimbatore. Lata M, AC Indore, Coimbatore. Madan K, AC Indore, Coimbatore. Malavika V, AC Indore, Coimbatore. Mary Lisha J. Mukiri Chinna, AC Indore, Coimbatore. Mummasani Asita, AC Indore, Coimbatore. Murukubel K, AC Indore, Coimbatore. Muthu R, AC Indore, Coimbatore. Naveen Kumar T. Naveen Kumar T. AC Indore, Coimbatore. Nilavarasi, AC Indore, Madurai. Pandithurai from Community Science College and Research Institute, Madurai. Parveen Banu, AC Indore, Madurai. Purushothaman J. Purushothaman J. Horticulture College and Research Institute, Coimbatore. Rajashekhar R. Agriculture College and Research Institute, Coimbatore. Sandeep G. Horticulture College and Research Institute, Perikulam. Silambarasan of Agriculture College and Research Institute, Madurai. Sivashakti T. Community Science College and Research Institute, Madurai. Shivashankar M. Agriculture College and Research Institute, Coimbatore. Sukanya K. Agriculture College and Research Institute, Madurai. Suresh L. Agriculture College and Research Institute, Coimbatore. Sweta AC Indore, Coimbatore. Tamil Amadam of AC Indore, TNA, Coimbatore. Venkatasamy, Agriculture Engineering College and Research Institute, Coimbatore. Vignesh P. Agriculture College and Research Institute, Coimbatore. Binita N. AC Indore, Madurai. Yama Santoshi Lavanya. Yama Santoshi Lavanya, AC Indore, Coimbatore. Yasmin of Madurai campus. Yasmin. and Yalini, Agriculture College and Research Institute, Coimbatore. Now the exhibitors and the presenters of the innovations are called. There are 20 presenters on the innovation. Number 1 is Oroka Infant Paul of Agriculture College and Research Institute, Coimbatore. The innovators will be given a certificate along with the memento. Smartwatch. Along with the certificate, they are also given away with a smartwatch to work more diligently and hardly. Followed by Atira BB of Agriculture Engineering College and Research Institute. Chitra R, Agriculture College and Research Institute, Coimbatore. Divi Bharati, Agriculture College and Research Institute, Coimbatore. Jagdish M. Jagdish M of Agriculture College and Research Institute, Coimbatore. Janani T, Horticulture College and Research Institute, Perikulam. Jairaj M, Agriculture College and Research Institute, Madurai. Komanaboina Raju Yadav of Agriculture College, Agriculture Engineering College and Research Institute, Coimbatore. Krishnaraj M of Agriculture College and Research Institute, Madurai. Mokita P, Agriculture College and Research Institute, Coimbatore. Followed by Niveda, Agriculture College and Research Institute, Coimbatore. Rahul Dev, AS, Agriculture College and Research Institute, Coimbatore. Ravori Sai Prasad, Agriculture Engineering College and Research Institute. Shaliga, Agriculture College and Research Institute, Coimbatore. Shanmugopriya S, Agriculture College and Research Institute, Coimbatore. Shanmugopriya to receive the certificate, followed by Sindhu Bharati, Horticulture College and Research Institute, Perikulam. Shivadarshan Priya, Agriculture College and Research Institute, Madurai. Shivadarshan Priya, Agriculture College and Research Institute, Madurai. Shivashankari Yel, Agriculture College and Research Institute, Coimbatore. Swatika, Agriculture College and Research Institute, Coimbatore. And Yogeshwari R, Community Science College and Research Institute, Madurai. Now it's time for the student volunteers who came up with a heavily working in day in, day out, along with the deans to make this function very memorable. The student volunteers can receive the certificates now. Mr. P. Davidson Rockins, 3rd PhD Agriculture Microbiology, TNAU. Followed by Mr. P. Godson Rockins. 2 Rockins are here. Davidson Rockins, 3rd PhD Microbiology, and Godson Rockins. The twins are getting the twins are getting the certificates. Mr. P. Jagdishwaran, 3rd PhD Agriculture Economics, TNA, Coimbatore. Mr. G. Karthikeyan, 3rd PhD Environmental Sciences, TNA, Coimbatore. Ms. S. Keertana, 3rd PhD Environmental Sciences. Ms. R. Sauntarya, 3rd PhD Entomology, Coimbatore. Mr. R. Muthu, 3rd PhD Entomology, TNA, Coimbatore. And Mr. D. Pradeep, 3rd PhD Nanoscience and Technology, TNA, Coimbatore. Organizing secretaries, Dr. N.O. Gopal, the professor working in Office of the Dean, School of Postgraduate Studies, will be given away with the certificates and a memento. And Dr. J. Venkat Prabhu, it's me. This Engineer G. Vanita, Assistant Professor, Office of the Dean, School of Postgraduate Studies, to receive the certificate. Dr. R. Prabhu, teaching assistant working in the office of the Dean, School of Postgraduate Studies. Dr. S. Ganesh, the teaching assistant, office of the Dean, School of Postgraduate Studies. And Mr. E. Sridhar, working in the office of the Dean, School of Postgraduate Studies. He is a teaching assistant working in the office. Now we stand flanking the Vice Chancellor and the Dean, Postgraduate Studies to have a group photograph. one announcement, please. All the outstation students are requested to go to Dean's office.
Super. It's me.
So the session is adjourned.